we share our screen to get our presentation started. So sorry about the technical difficulties. It's not okay. So we are here for 3 hours, and I would suggest making sure you have something to drink and maybe some snacks. And let's make sure everything looks good. All right, hopefully you can see my screen and hopefully you can hear me as well. Can everyone hear me? Yes, thank you, Marianne. You are wonderful. Okay, let's do this then. If you are not already following Fiveable, make sure you go to your socials and follow Fiveable at Think Fiveable. Yes, hi. We're going to do introductions in just a minute here. If you are hoping to study for AP Biology, this is the place. So hopefully you're in the right spot. We are at night 2, and we will be covering units 5 through 8. And we'll go into exactly what those units are a little bit. And we have 3 student hosts with us tonight. Again, I said my name is Kari Parnan. I'm your teacher host for tonight. And then if I could just have my student hosts hop on and introduce yourselves real quick for our audience.
I can go first. Hi, everyone. My name is Madison, and I am a high school sophomore in Illinois. And the questions we're actually asking to us student hosts is, what is your favorite superpower? Personally, I would say telekinesis. I feel like it'd be really cool to just be able to move like objects without actually like physically touching them. That's what I just personally think. Yeah, so excited to see you, everyone.
Hey everyone, I'm Samhita. I'm a high school senior from Texas. And I would say my favorite superpower would be time travel.
Hey everyone, I'm Jeffrey, and I'm from Minnesota, and I'm a sophomore in high school. And my favorite superpower is invisibility, I guess, because it's more freedom. I don't know.
Awesome, love it. I didn't even think about the answer to this question, so I'm going to have to get back to everybody. But it's a good question. Thank you for introducing yourselves. I'm so excited that we have student hosts. They're going to be here helping present with me tonight. They're going to be watching the chat. They're going to be looking at the Q&A. So right now I'd like to point out, I know a lot of you are already in the chat. That's probably the default setting. But there's also right below the chat button on the right side of your screen, there's a question mark symbol. So that is where you can ask specific questions. And then throughout the night, if you see someone else ask a question similar to it, you can vote up on it so that it becomes more likely to be answered. And then if there's anything we don't get to tonight in terms of answering your questions, we will build them into the next 2 evenings presentations. So we'll look at, hey, everyone's really freaking out about this one topic. We should really cover a little bit more tomorrow night. So use the Q&A, and then our student hosts will be monitoring both of those for you. Our cram schedule right here, May 5th was yesterday. So if you didn't catch that, there's a little green bar at the bottom of your screen that has the AP Bio Cram Hub. So that's where you would find slides and replays in the review packet that we made for you. And then tonight, again, is May 6th, units 5 through 8. We're going to be covering genetics, evolution, and ecology. Tomorrow night is going to be science practices 1 through 3. And I'll be covering that with you. So I'm really excited about that. And then May 8th will be practices 4 through 6. So the math stuff, the graphing, the drawing conclusions. And then May 9th will be the grand finale where our student hosts will be taking it over and covering units 1 through 8, giving you their test taking strategies. And we'll probably also cover some Q&A moments as well. This is our official agenda for tonight. So we're going to do a quick welcome and check-in. I'll give you an overview of the 4 units we'll cover. And then as it says there, we're going to cover each one at a time. We'll have a short break in between each unit because we're going to be here for 3 hours. And I'm a human and you're a human. And for sure, we need to stand up and take a break, make sure we have some water and some snacks and just, give our brains a minute because I know this is tough content. Okay, Madison, do you want to jump in and talk about how to make the most of a cram finale?
Yes. So now you may be wondering, after the first day of your cram session, how can you make the most of these cram finales? The first thing will be taking notes. We believe that active listening is very important throughout these cram sessions as we're giving you a load of information. However, we do not expect you guys or recommend to you guys to write down everything. That can end up with you actually not attaining as much information as you would have by just active listening throughout the session. And also, besides the Fiveable community, the Cram Hub provides so much resources for you. We have slides, review packets, and replays just for you guys. So be sure to follow along by looking at these resources as well as our replays if you have missed some of the sessions. And we also have a group chat that can be used for sharing tips, tricks, and participating when we ask questions. And lastly, we do have a Q&A tab, like Ms. Kari said, and be sure that if you guys have any questions, be sure to look at it first at the Q&A tab to see if your question was answered or has been asked already. And yes, we'll be taking breaks. We'll be taking breaks throughout the units, so do not fret.
Mhm.
And here are some of our community agreements. First of all, we do ask for you guys to be supportive. While Fiveable is interested in having an engaging community, we also seek for a supportive and kind community. So let's lift each other up throughout this hard time of understanding all these terms and understanding all these types of topics we'll be learning today. Next, be patient. Some of you guys are very interested in just grinding throughout all the units. However, we are very interested in just laying out some info without giving you too much pressure for the upcoming exam. So we do have 5 nights. And if you have missed any, once again, you're more than able to look at the replays. And lastly, like I said before, be kind. Besides the support of the community, we are asking for a community full of kindness and not really bringing much negativity. So we do recommend having a positive community throughout this time being. Before we get started, you guys have a chance to win $100 by sharing your own cram experience on Instagram or TikTok by using the hashtag FiveableCram and tagging at ThinkFiveable in the post or caption. You are able to be selected as a winner throughout the 2 weeks of cram finals and winning $100 by making us laugh or getting a ton of views and influence.
All right, thanks, Madison. Appreciate that. So I'll give you a second. Just look at your screen if you want to take a picture of that slide so you don't forget to try to win $100. You never know. So take a picture of it with the directions and maybe do that while you're watching tonight or after the cram or at some point in the next few days. So I'll give you a second to take a picture. Wonderful. All right. It's 7:12 and it looks like we have a really good group of students here. We have about 250 students joining us. And I just wanted to give you an introduction about me. My name is Kari Parnan. I really hope some of my students are attending since I told them to. So hopefully they're active in the chat and happy to spend the time with them as well as you. I have 3 little kids. They're actually older than that picture, but I couldn't find another picture of the whole family. We live in Michigan, West Michigan to be exact. So it is 7:12 here. I know that it's not 7:12 where you live necessarily, but I am a science teacher and originally from Illinois. I went to the University of Illinois at Urbana-Champaign, and I just love I love science, obviously, but I'm really excited. I love live teaching. I've been with Fiveable for several years, and I'm just loving this new way of having students also helping because they can engage you in different ways that maybe I couldn't. So I'm really excited about that. All right, Madison, ready?
Perfect. So let's get started by asking you guys, where are you guys joining us from? Be sure to let us know in the chat.
I love this part. This is one of my favorite parts to see where people are joining us from.
I see a lot of New Yorks.
New York's nice. Oh my gosh, it's so many. It's so fast. 80 messages. Holy moly. Canada, Michigan, let's go. Okay, I love it. I love it.
Great. Let's move on to the next slide as we keep continuing and adding comments to the chat. But we're going to move on to our next question. Respond to the poll that is below the Q&A section, and it'll be asking you which topic do you feel most confident in throughout the AP Biology course. So I'm gonna actually answer this for myself as I see the poll coming up. Personally, I was really confident in regulation of gene expression. I believe based on my class, did a lot of projects and activities and more specifically group activities on this unit. I feel like I was more confident, but I actually want to ask Hamita, our other student host today, on actually her answer for this question.
For me, it's definitely evolution. It's my favorite topic, and I feel like it was pretty easy.
I agree. That was always my favorite too. Based on some of the answers to this poll, it's looking like regulation of gene expression is our area where we feel least confident. So I want you all to know we are going to focus on that tonight. We're going to focus on it all, but as always, if there's anything we're not quite digging into that you just really, really need help with, make sure you put that in the Q&A so that we can create something that will be especially helpful for you in our next live stream.
Great. So we're going to go quickly show you an overview of tonight for units 5 to 8. So throughout these 4 units, we're going to go through a lot of information and topics. However, we'll be mainly heavily focusing on the topics highlighted. So if we are continuing on with the session and you guys are not understanding as much throughout the other topics, you are welcome to ask it in the comment section or look at the other resources Fiveable provides you regarding these topics as well. Now, without further ado, I will move it over to Samita to introduce our first unit we'll be discussing, which is hereditary.
All right, our first unit, it can cover between 8 to 11% of the AP exams, so you got this. And these are the skills that will mainly be addressed in nights 3 and 4. But just check if you know of these skills, and if you need more practice, you can always go back and relearn it. And so we won't be going through all of the vocabulary right now, but if you all see something that you know of, then you're good to go. But if you see something that feels a bit confusing or you're still iffy on it, then it's a good idea to go back and review it.
Okay, thanks everybody. You're awesome. So let's just start off with the beginning of Unit 5, so meiosis. And depending on when your teacher covered meiosis, it could have been a month ago, it could have been 5 months ago. And I want you to, don't get too freaked out about this diagram, but look at that question at the top where it says, why meiosis? Think about that. What's the purpose of meiosis? It's this process, it happens in cells, what's going on? So just take a moment and throw it out there. What is the purpose of meiosis? Not the steps, just the purpose. What is the purpose of meiosis? Okay, I'm seeing some genetic diversity in reproduction. I see the word gametes. Love it. Gametes. Okay, good, good, good. So yes, absolutely. I love all of your answers. Oh, genetic diversity. Yes, we're going to get to that. Excellent, excellent. So meiosis, and again, you can't really see in this diagram. It's pretty small, and we're not going to go through all the steps of meiosis because honestly, according to the College Board, that's not something you need to know. You don't need to know the steps of meiosis. You likely were taught all the steps of meiosis so that you could track those moments where really important things happen. And I'm going to talk about that right now. So I believe in the review packet, there's a chart for mitosis versus meiosis. And this is one of the things that you definitely need to know. So mitosis is different than meiosis. Mitosis is where we are just creating identical cells. So you're replicating somatic cells. Somatic means body cells or non-sex cells, okay? So any other cell in a body that are not related to reproduction, like sperm and egg, okay? Mitosis is also where we see asexual reproduction, right? Because if something is reproducing asexually, they don't need gametes. So just like a bacteria can just double its DNA and split in two, that binary fission thing, remember? That is going to be mitosis as well. For us, it's happening when we get a cut, right? Or when we're growing, right? We need to make new cells. Meiosis, all right, here are the clear differences. We're creating gametes, which means sex cells. And then we are also creating haploid cells. They have half the DNA, half the chromosomes, not like it's randomly cut in half, right? So instead of being 2 copies of every chromosome, we now have just 1 copy of every chromosome. And then the other important thing to make sure you know is that daughter cells are genetically unique, right? So it's not going to be a blueprint. It's not going to be a carbon copy. It's not perfect DNA replication, right? That's happening in mitosis. They should actually be unique. So that is important as well. One of you in the chat talked about meiosis generating genetic diversity. So I just want you to think, well, how? Right? Yeah, it's making 4 cells, but well, aren't they just the DNA cutting it? What is happening to make genetic diversity? And you can either answer in the chat or you can just think about it. I see someone asking a question about, do you need to know the steps of mitosis? You don't need to know steps necessarily, but just like with meiosis, you need to know kind of what are the chromosomes doing, right? So like, are homologous chromosomes separating? Is it your sister chromatids separating? And things like that would be important to know. Okay, so to answer the question, how does meiosis generate genetic diversity? One of the ways is crossing over. So this image is showing homologous pairs of chromosomes. And so what I want to show you, let me see if this will let me do it. Hold on one moment. Okay, it's not going to let me do my little pointer. But the blue chromosome and the red chromosome, those are representing maternal and paternal chromosomes. So if you remember karyotyping and you looked at the human set of chromosomes 1 through 23, right? There were 2 number 1s, 2 number 2s, 2 number 3s, so on and so forth. So these are that same thing. This would be like 2 of chromosome 1 next to each other. The reason why it looks like a butterfly is that it has already replicated, right? So that whole blue thing is a duplicate, right? So you've got the little butterfly wings, right? They're sister chromatids. They are identical. But the homologous chromosomes from maternal and paternal cross over, and then you get a completely different set of alleles, right? You might have a dominant allele crossed over with a recessive allele. And so we're kind of changing up the possibilities of what traits are possible for the next generation. The other way that genetic diversity happens is through independent assortment. And what that basically says, if you can look at this picture, it's how the homologous pairs line up during metaphase at the metaphase plate. The first one is showing all blue on the left, all red on the right. And then all the other ones are the different combinations. And this is just for something with 3 sets of chromosomes, or 3 chromosomes, not 3 sets, 3 chromosomes. And even just from this small section, there's 8 possibilities here. So with us, we have so many more possibilities. It's 2 to the n, so 2 to the 23rd power possibilities of how the chromosomes will align. So that's another way that genetic diversity occurs. Something not on the slide is that random fertilization is the other thing too. So like the random 1 egg that's fertilized by the random 1 sperm, that creates genetic diversity as well, because every single sperm and every single egg cell, or the gametes, are genetically unique. So this question asks, does this happen in asexual reproduction? So does this happen in asexual reproduction? I'm going to go ahead and make a poll for that. So that we can get used to this poll situation. So yes or no, does meiosis happen? Or even just crossing over, independent assortment, does that happen in asexually reproducing organisms? Way to get on the vote. Good job, everyone. Wonderful. Okay. Yes, you're wonderful. It is no. The answer is no. It does not happen in asexually reproducing organisms like bacteria, for example. They can get genetic diversity other ways. That's not something you need to know, but they can. They have other ways that they can get genetic diversity where 2 bacteria can kind of come next to each other and swap DNA. But that's not necessarily something that you need to know for this course. But if you take biology in college and higher level, then you probably would. Okay, I'm going to hop off to let Madison take you on to the next part.
Perfect. So kind of as Ms. Carter said earlier, we kind of discussed a little bit on genetic diversity, but what is genetic diversity, you may be asking? Well, genetic diversity kind of discusses on the variety of genetic combinations within a species. So, for example, corn. Corn doesn't have just a specific genetic format. There's many different sizes and colors and tastes of corn, and that itself can be a very good representation of what genetic diversity can hold. Now, understanding genetic diversity, you guys could start responding to this poll on why is it important?
Poll's coming. Sorry. There we go.
I see you guys. I see a lot of you guys are answering correctly, and yes, it is. It is B. It allows for species to be resilient to environmental changes. So, kind of following with what Ms. Kari is discussing on mitosis and like the cell divisions, mistakes do happen. And if a spinal fiber does break and does not separate the sister chromatids, do you guys know what that is called? So yes, it is non-disjunction. And non-disjunction is when the pairs of homologous chromosomes do not separate normally during cell division. And now I'll be asking you guys another question. What is the outcome of non-disjunction? I'll answer it here, but you guys are more than happy to leave it in the chat. Non-disjunction, my apologies, does have outcomes varying in many birth defects that can happen. As you can see here with the karyotype. You do see that while there's only should be 2 chromatids within a pair of chromosomes, in one of the actual areas of chromosomes, there's actually 3 pairs instead of 2, leading to a genetic disorder. Great. So on the next slide, we'll be connecting meiosis to genetics. So I'll give you guys about 2 minutes to answer this quick right. To summarize in one sentence, what is Mendelian genetics, autosomal dominance, dihybrid cross, and the 9:3:1 ratio? I'll give you guys once again 2 minutes. So we'll come back together at whatever time it is. For me, it's 6 o'clock. So for me, 6:29. Whether that be 8:29, 7:29 for you guys. So I'll be back in 2 minutes. Great. I'll go now quickly summarize the sentences. So Mendelian genetics is the pattern of inheritance explained by George Mendel that actually used pea plants for his theory, which if you guys were wondering why he did use pea plants, that was actually because they're more easier to actually be moved, and they were not as difficult to manage as it would be with other specific plants. And further, the genetics actually discusses on the patterns of how traits are passed on from parents to offspring. Next, autosomal dominance is where and the traits are passed down from the parent to the child. So alleles for a gene located in a non-sex chromosome. Next, dihybrid crosses are genetic crosses that are between two individuals with two traits controlled by two genes. For example, you see here on the left, there's a chart showing the actual like Punnett square, which is the cross between two individuals with two traits controlled by two genes. So it's between two Ps. However, it is varied between its size and color. And instead, a monohybrid cross would then be just two specific traits. A specific trait instead. So for this case, it would just be either the sizes of the Ps or the color. And lastly, the 9:3:1 ratio is the phenotypic frequencies predicted in some dihybrid crosses.
Okay, thanks, Madison. Let me just try to get myself back down over here. Could someone do that for me? Move my, thank you. Okay. All right, thank you so much, Madison. I know this is big stuff, it's heavy stuff, but I want to let you all know you're doing so well. I love how active you're being. You're answering the polls, you're asking good questions. I'm already looking at the Q&A. So just know that the fact that you're here, it is Saturday night, and you are choosing to be here right now, that's huge. And the exam's not till Wednesday, right? And the fact that you're here, you are going to pick up some stuff, and you are going to feel more confident that you took this time and you made this happen for yourself. So just be really proud of yourself for even being here tonight. And again, just some reminders, you are in charge of your own brain, and you know when you need a break, you know when you're distracted and maybe something's distracting you, and you know to put that away. But I'm so happy that you're all here, and I'm so proud and so excited to, I would love to just know how the exam goes for you on Wednesday. So anyway, real quick practice thought. I don't think that there's a poll for this, and no need to write about it in the chat. But if we're talking about PPODs being inherited through autosomal dominance, autosomal just means that it is on non-sex chromosomes, so not the X's or the Y. Okay, so if it's inherited through autosomal dominance, and green variation is dominant to yellow, we have this cross of a green and a yellow plant, and their offspring, we have 13 green and 15 yellow. So just think for a moment, what were the parent genotypes? So it's one trait, two variations, green or yellow. So take a moment and just think about that. All right, so hopefully you were thinking, well, if one parent is green and one is yellow, and we know green is dominant, but they were still able to have some recessive offspring, then one of the parents must have been heterozygous. And so if you look at that cross right there, I've got my big G allele and little g allele representing one of the parents, and then the other yellow plant would have been little g, and that would give us about a 50% outcome of green versus yellow. And even though it's not perfect, right? Like obviously 13 and 15 are not even splits, but we should remember in genetics that these are all hypothetical, right? In terms of, oh yeah, we would expect 50%, but that doesn't necessarily happen. If you think about humans and babies, there's a 50% chance that your baby's born XX chromosome, and there's a 50% chance that it's XY chromosome, right? And every single baby, it's a brand new chance. I know my sister had 3 daughters in a row until she had a baby born XY. And so the real life outcome is sometimes different than what you would expect. So there I just wrote, each parent must have at least one recessive allele for an offspring to also show the recessive phenotype. And remember, phenotype just means that physical trait that we are seeing, whereas the genotype is going to be what alleles are we talking about. So the parent alleles or genotype are shown there as well, heterozygous by homozygous recessive. Okay, so this one right here is talking about expected versus observed ratios. You'll get more into this in 2 nights where you do a little bit more math, but I just wanted to talk you through it. So if we do a dihybrid cross, that means dihybrid, meaning we're looking at two traits. We're looking at fur color and then also fur texture, so rough or smooth. And what I want you to think about, and we're not going to go like give you a bunch of time to solve this. I want you to think about how you would solve this. Think about big B, big R, little r. And then the other parent is big B, big R, little r. I'm curious to see how you would solve this because there are two possibilities and they're both right. Yes. So I see people saying dihybrid cross. I see someone saying foil. I love the foil method. Good. Awesome. Okay, so I see a lot of you already jumping in right away, and you remember what Madison said about the 9:3:3:1 ratio, and you're absolutely right. That's what we would expect to see. And sometimes when I talk about 9:3:3:1, and then I haven't talked to my students about it in a while, they're like, "What are you talking about?" But if it's a dihybrid cross, we would have 4 alleles on top, 4 alleles this way, right? And so we'd have 16 boxes that are going to be filling our possible genotypes. So 9 means 9 out of the 16 possibilities would likely come out black rough. So dominant, both dominant phenotypes possible. So 9 out of 16. The 3 and 3 are, one dominant trait, one recessive trait shows up. The other 3 is one recessive, one dominant. And then finally, that 1 out of 16 is your recessive. So we'd be looking for little b, little r, which makes sense that it's a 1 out of a 16 chance for a cross like that. I believe Ms. Shoemate is going to be going over the actual probability of doing little crosses in 2 nights. So if that's something that you're like, oh, I really wish I had more time, then just sit tight and keep coming to our sessions and you will get what you need to know. Awesome. Just a quick plug for the math session on Monday night. If observed results don't quite match the expected ratio, right? So if you've got these guinea pigs and you're like, oh my gosh, genetics, I learned about this, it should be 9:3:3:1. And then all of a sudden you look at the outcome and you're like, whoa, there are so many of these guinea pigs who are showing both recessive traits. Like, how is that even possible? And so that's when you could be super dorky like me and throw in a chi-square test and see if your results are statistically significant. Because maybe something's going on. Maybe the genes are linked, right? Maybe those 2 genes, the white smooth genes and the black rough genes, maybe they're linked. And so when crossing over happened, they didn't actually they didn't sort independently because they're on the same chromosome. Okay, when we do Punnett squares, when we separate those letters B and R, we don't put them next to each other. We separate them because we are assuming independent assortment. We're assuming they separate, but I'll go more into linked genes tomorrow night as well. If you are like, what is she talking about with linked genes? Woo, we are good. Okay, take a minute and take a breath. How's your brain feeling so far? I said, don't be afraid to ask questions. This is how we learn. And just take a moment because you just listened for 30 minutes straight, which is a really long time. So I'm actually going to throw in a impromptu break for everybody. We only have a few more slides left on genetics. So just take a moment, take a little breath. Oh, someone just worked 9 hours. Oh no, that's so tough. So Liliana, chi-square is going to be covered in 2 nights. So not tomorrow night, but the next night. Yay, I see someone saying they're less worried now. That's so wonderful. Yay, yay, yay. Hardy Weinberg, the math section of it will be covered on night 4 as well. And pedigrees, we're going over tomorrow. Yay. So for those of you who maybe missed like the intro of what night was, maybe I went too fast. Tomorrow, I'm going to be covering concept explanation. So basically, I'll take all sorts of different concepts from pedigrees, from basically any topic we've covered, and we'll just say, hey, can we explain it? How do we explain it? We're also going to be looking at visual explanations. So we'll look at any kind of FRQ or multiple choice that has an image with it, right? They might give you like a whole picture of carbon cycling or something like that. And we're just going to go over how do we answer that. And then of course we'll do, tips and tricks of like day of, right? Like on the day of the test, how do we sorry, 1 second, said. Okay, we'll go over like day of, how do we do? What do we do on the day of? So awesome. All right. Hopefully you're all coming back. I just have a few more slides before we're going to jump into Unit 6. So get yourself prepared. I know a lot of you were talking about Unit 6, like that's the least confident. So we are going to try our very, very best to get you confident. And yes, anyone asking about math stuff, it's all going to be happening on Monday night. Okay. So math and graphing, come Monday night and watch that stream. And I know that you will feel better. I promise. Okay. All right, let's quickly finish up genetics. We've got to talk about non-Mendelian inheritance because you spend a lot of time learning about things that are so predictable, and you learn about, you probably learned about it in middle school even, right? Like dominant, recessive, all that. But in reality, how traits are passed down are so complicated. And so most inheritance patterns are not Mendelian. They're deviations. They're different, right? Like skin color, even eye color, hair color. It's not so simple as dominant and recessive. If you remember from the very first picture I showed of my family, right? Like I have blonde hair, my husband has black hair, and our kids have very unique hair and eye color as well, right? So it's not so simple as, actually both of my boys have blonde hair, which you wouldn't think based on your old knowledge of dominant or recessive. So things like codominance, multiple alleles, mitochondrial. The reason why I don't have definitions for these is that you're not going to be asked to define them, but you might be given some information about, mitochondrial inheritance. And the thing you'd have to put together is that, oh, that's maternal. If it's a mitochondrial trait or mutation, organisms get their mitochondria from their mother because the egg has mitochondria in it, whereas the sperm doesn't, right? The sperm does not have a mitochondria. It's not going to be from the dad. Yeah. Sex linked real quick. I'll just give you a quick example so we can talk about that. But sex linked generally is going to be talking about traits that are on the X chromosome. It's pretty rare to have a Y-linked trait. If you've ever looked at a karyotype, the Y chromosome is like really small. There's not a lot of genes on the Y chromosome. So generally we're talking about stuff that is on the X chromosome, which makes for a really unique situation. We talk about traits being passed down. If you are XY and you're born with an XY chromosome, you only have one X. So if your mom has some sort of like disease that's X chromosome based, that's really rough for you, right? It's more likely gonna harm you. So this quick example talks about hemophilia. The mother has 2 normal alleles. The dad, sorry, where am I? Father has hemophilia. There it is. So because it mentioned in this slide that hemophilia is a sex-linked recessive disorder, that means to have the disorder, you'd have that recessive allele. And as male, you only have one X chromosome. So if there's one X little h, then you have it. So if we look here, what are the probabilities of the sons getting it? You can see right there, thankfully, because mom had 2 non-hemophilia or normal alleles, her sons only got their X allele, X chromosomes from her. So there's actually a 0% chance that this family does have hemophilia. We could do a whole pedigree on the next generation, right? Because now the daughter's a carrier, so she could potentially pass on her chromosomes to her son. Alrighty, yay. So now we are gonna do some practice questions. So I'm gonna share a different screen with you. We're doing something different. So just give me a moment. We have some practice questions for you. So get your phone out because you're gonna need to take a picture of a QR code. This is the first time I've done this, so hopefully it is okay. All right. Nope, we need a Chrome tab. All right, so this is just going to be covering Unit 5 so far. okay, why do I still see my face? I want to see my screen. There it is. Okay. I was like, what the heck? All righty. So I'm going to hit start quiz and we're just going to kind of wing it and see how it goes. Okay. There we go. Start quiz. So take a picture of that QR code because I don't know what happens if I hit start. If I hit start, it might, I don't know what's going to happen. So take a moment, get that QR code, and then I'll hit start. Okay, we'll see. I have 70 of you, 80 of you. Let's go. So once you join, I'll hit start, and then I know we're kind of like running low on time, so I might not have us answer all the questions so that we can get to Unit 6, okay? All right, let's see what happens. All right, I'm just gonna reveal the answer. Yay! 83% of you said gametes. So wonderful, wonderful job. Next question. Go ahead and try question 2. You got this. What do gametes have? Woo! Haploid chromosomes. Good job. Awesome, awesome. Okay, good. So independent assortment, I mentioned that earlier. Yes, 99%. Amazing. Segregate independently of other allele pairs. I'm going to go 2 more questions and then we will. Wonderful. Increased genetic variation. Amazing job. We'll do 1 more and then we'll move on to Unit 6. Amazing. 99% again. I'm so proud of all of you. That's so good. Wonderful, wonderful. Okay, non-disjunction. That's absolutely right. Love it. Okay, let's move on to Unit 6. Let me get my slides back up here. And since we kind of took a short break a few moments ago, wait a minute. Hold on. Sorry. I need to be in presenter view. I don't like this thing. We already took a short-ish break, so I'm going to skip this one. Why? Okay. My Google Slides are taking forever. I apologize. Oh, brother. Okay. So, guys, we're just going to skip that 60-second break because I think you are all going to be just fine. All right. So I'm going to take a little break and hand it over to Sabita. And then if someone wants to move me to the wing, since I don't know how to do that. All right. It's all yours.
Okay, so we'll be going to our second unit, which is gene expression and regulation. And this is actually 12 to 16% of the exam. And just like the previous unit, these are all the skills that we'll be practicing on nights 3 and 4. So like I said before, if you know some of these, then good, you're good to go. If you're still feeling a bit iffy or you're not as confident on it, then I strongly suggest that you go back and review it. Yeah, and this is these are the vocabulary for this unit. They usually don't test you on the vocabulary, but it is good to know for just for your understanding.
Oh, it's me. Sorry. How do I get myself back? Can someone do it for me? Okay. My student host will help me. There we go. Oh, goodness. If my students from Michigan are watching, I hope you're laughing at me right now because you know that I am not the best at these sort of internet things. So anyway, so I thank you so much for my student hosts who can help me. All right, so in Unit 6, these are just kind of the big things, right? DNA replication, protein synthesis, regulation of gene expression, mutations, and biotechnology. Now, because a lot of you are saying this is your, least comfortable, least confident unit, I'm going to go over all that. Okay, so instead of like the other unit where I kind of just chose 1 or 2 big things, we really are going to touch on all these things. Okay, and we're going to do great. So let's start by just knowing DNA structure. I know that Mrs. Shoemate covered this yesterday because DNA is a macromolecule, right? A nucleic acid is a macromolecule. Uh-oh, why did my face get so big? Thank you. So DNA is a macromolecule, but when we think about the structure of DNA, you maybe learned about it in 5th, 6th grade. You learned that it was a double helix. You learned about maybe that it had bases that paired. But here's where I'm going to elevate and ask you a question. How does the structure of DNA compare between prokaryotic cells and eukaryotic cells? So take a moment to think about it. If you're like, what's a prokaryotic cell? What's a eukaryotic cell? That's okay. Prokaryotic cells are going to be your cells that don't have a nucleus, and eukaryotic cells are like you and I. Eukaryotic is like you, and you are a human, and you have, you are a multicellular organism, and you have a nucleus in each of your cells. There are some single cell eukaryotic organisms, just so you know. So don't think that it's just only multicellular. But big picture, the difference between a eukaryotic and a prokaryotic is, well, eukaryotic cells have their nuclei holding the DNA. But what I wrote there is prokaryotics typically have circular chromosomes, like you see in that picture there. And then eukaryotic cells, we have linear chromosomes, right? We've seen them in a karyotype lined up all nice. We have linear chromosomes. They both can contain plasmids, which are just small circular pieces of DNA. And we will talk about bacterial plasmids in a little bit when we get to biotechnology. The other things about DNA structure that should be reviewed if you attended last night is knowing what the building block of DNA is. So this is actually my hand-drawn artistry of the building block of DNA. So just take a moment and tell us, what is that building block of a nucleic acid? Yep. Okay, so hopefully you said a nucleotide. All right, so when I put a little purple box around the one nucleotide, the nucleotide is the building block of the DNA molecule, and it has a phosphate group, a sugar, and a base. And so phosphate, sugar, base. I don't even know how old you all were when the All About That Base song came on. It was popular, but I happened to be teaching AP biology when it was popular. And so I would tell students to kind of sing phosphate, sugar, base, kind of like All About That Base, because then it just helped them remember that, like, oh, the phosphate's out. I'm kind of, the phosphate and sugar are together because that's your backbone, and then your base is the middle. Some other key characteristics of DNA that just, again, review: the hydrogen bonds are what are between the bases. Okay. Now, hydrogen bonds are not like super strong because we do need to open up the DNA structure from time to time, right? When we're replicating it or transcribing it. So hydrogen bonds are really cool, really nice in the middle. The other thing, anti-parallel strands. So that just means they're upside down from one another. So if you look at my drawing, the phosphate group, which is the P with the 4 O's around it, that's on the top. But then if you look at the other strand, the phosphate group's on the bottom. So they're anti-parallel. They're kind of upside down from one another. You should know that A and T bond and G and C bond together, and that we've got a phosphate sugar backbone, which is covalently bonded, which is stronger, which is good, because if we've got enzymes messing around and opening things up, we don't want our whole DNA molecule to completely disintegrate. We need it to be somewhat of a strong structure, right? It's pretty important. It has the information to make us who we are. Where DNA and DNA replication fit in, if you remember the cell cycle, again, you would have gone over this yesterday. It's a part of Unit 4. We talk about just the life of a cell, right? The cell cycle is the life of a cell. So you're sometimes making more of yourself and sometimes you're not. Sometimes you're in the G0 phase where you're just doing your job. You're not needing to make more of yourself. You're just doing your job. But if you are a cell that is about to need to do mitosis, you need to replicate your DNA. So when does replication normally occur in the cell cycle? Sorry, I don't have a poll for this one, but you can just type it in the chat. So when does DNA replication happen in this cell cycle? So I get a cut on my skin. I need to make more skin cells. Oh, shoot, what do I do? Oh, yay. Yes, S phase, synthesis. Lovely. Why does it occur? I kind of gave that away a second ago. Yeah, in preparation for cell division, right? So we might need new cells because of growth, right? So like my children, they're growing, right? So like they're constantly making new cells because they're getting bigger every single day. But then also sometimes repair, right? Like a week ago, my son jumped in the grass barefoot and landed on this like crazy sharp animal bone and tore like the bottom of his foot, had to get 5 stitches. And it's been only 9 days, 10 days, and he played soccer today, like on his feet, right? Because his body was going through crazy mitosis and making more and more cells so that he could restore and do well. Amazing. And then also asexual reproduction, right? So mitosis isn't just about our cells. It's about some organisms just do mitosis to make more of themselves and they replicate. Yeah, I know. His foot, you guys, it was awful. It was just terrible. It was the worst thing. I can't even imagine. I don't even want anyone touching the bottom of my foot. And he had this giant bone that hurt him. So anyway, moving on. So when we talk about DNA replication, I have really good news for you that you don't need to memorize like the steps of DNA replication, but what you're expected to know is the enzymes and kind of just big picture, right? So I wrote a couple of things here, but knowing that it's replicating in a 5-3 style. So it is going in a specific direction. It can't just start on one end and go on both strands, replicate them. That's why they're going in separate ways, right? Because we go 5-3 this way and 5-3 this way. Here are some of the enzymes. We've got helicase, which unwinds the helix. We have topoisomerase, which prevents supercoiling. Because if you think about, like, if you've ever had like a string that is kind of braided, if I were to just start pulling at the string like this, it's going to supercoil at the bottom. Like I always say this to my students about like my hair dryer or my straighteners, they always get really coiled, right? And it's just like a big wiry mess. And so I think of the word topoisomerase is a mess. Like as a word, it's a messy word. Topoisomerase, what a mess. Like even just looking at it, it's such a mess. So think about this messy word is what's there to prevent supercoiling. It's to prevent like the giant mess. DNA polymerase is an easy one to remember because DNA is in the word and it's polymer, right? ACE. So it's an enzyme that makes polymers, right? Makes DNA into a polymer. And then ligase is the one that's going to be covalently bonding. Thank you. It's 8 o'clock. You're right. You're the best, everybody. Ligase is going to be the one that kind of seals everything up. It's kind of like the glue is what I would tell my students. All right. So I'm going to skip that and have you take a look at this diagram. And I'd like you, oh wait, not yet. The diagram's coming. When we have DNA replicating, you can see the top strand is getting new nucleotides in like a perfect straight line. Like everything's just good to go. And then it's going toward the replication bubble. It's following helicase. Whereas the other one on the bottom is going in the opposite direction, but the bubble keeps opening up to the right. So it's making new nucleotides to the left. And then all of a sudden, new nucleotides to the right are happening. So it's got to hop off, the DNA polymerase does, and it has to keep going over and over and over. So I'm going to make a poll because you do need to know these words, discontinuous and continuous. Hold on. I'm trying to do technology. Oh, just kidding. We don't need to do a poll. It's fine. So thinking about which one is discontinuous versus continuous, your top one is going to be a continuous strand where the other one is not, right? Because it's making those Okazaki fragments and has to continually catch back up to where things were separating. All right. 5 to 3 prime end. I have to tell you, when I was a student, this was the hardest thing for me to understand. I think it took me copying down and actually drawing out nucleotides over and over and over again for me to really understand what 5 to 3 prime meant. It really did. So if that's you, like dig into that confusion and figure it out, right? Like draw those nucleotides, draw out 2 strands and make sense of, okay, if it's going 5 to 3 prime, what does that mean? Because when you make sense of your own confusions, you will remember it so much better. So when I talk about DNA polymerase only being able to add nucleotides in the 5 to 3 prime direction, first let's look at this molecule in 5, find the 5 prime end. Your 5 prime end is always going to be connected to the phosphate group. And here's how I remember that. Phosphate, it doesn't start with an F, but it has the sound F. Phosphate, high five. Okay? So phosphate, high five, that's your 5 prime end. Your 3 prime end is going to be on the opposite. So when I say it can only add things in the 5 to 3 prime direction, it means the 5 prime end of a new nucleotide is added to the 3 prime end of an existing nucleotide. Okay, I'll say it again. The 5 prime end of the new nucleotide is added to the 3 prime end of the existing. So again, draw it out if you need to. Do it, because this stuff is hard, and you are all fantastic, okay? So I wrote it right there. Sorry, I forgot I had it on the slide. The 5 prime end of the new nucleotide is added to the 3 prime end. And that's just what DNA polymerase can do. That's all it can do. All right, take a moment. See if you can find out, based on what you know its job is, which one of those purple boxes is each of those enzymes. So the words are down here: helicase, topoisomerase, DNA polymerase, primase. Which one of those is each of these boxes? So just take a moment and do that in your head. Sorry, there was no way to really pull this one. And by the way, if you are taking the AP exam, don't like aggressively clap your hands when you're remembering 5 prime end phosphate. Be like, this lady told me to do it. Awesome. Oh, that's a good idea to use colors there. Love it. Okay, I already see some people getting the answer. So here we go. That pink one, correct, is topoisomerase. It's going to be ahead of helicase because that's where the supercoiling is happening. helicase is always an easy one, right? Because you're like, it's the thing that's unzipping the DNA, right? It's the helix, has the word helix in it, so it's breaking the helix. Primase is going to be that one in yellow because it has to lay down a primer first, because like I mentioned, DNA polymerase, it can't just start laying a nucleotide. It has to lay a nucleotide to a 3' end. So if there is no 3' end to add to, it has nowhere to go. So that's what's going on there, okay? And then DNA polymerase is the one, obviously, that's adding new nucleotides. It's that red enzyme. Back in the day, before they changed the AP or the bio exam, it used to have to memorize all. There were like 3 different types of DNA polymerase, and now you just need to know that there's one. I mean, there are more, but you just need to know the one DNA polymerase in general. So yeah, so again, you do not need to know the difference between DNA polymerase 1 and 2. Okay. Okay. We've got a student host coming up to talk about mutations for you all. So I will be quiet for a moment if they're still here. You ready to do mutations? Awesome. Go ahead.
Right, so mutations are just random errors that occur at the nucleotide level, right? So yeah, they could happen at any time, and sometimes mutations are good and sometimes they're bad. It's just force of nature, I guess. So can you identify the sources of errors in this DNA image? Okay, yeah, so according to this image, there are errors in the DNA replication. Basically, when the incoming UV photon hits the DNA, it basically loosens the bonds and weakens it, thus making the DNA more susceptible to mutations and creating errors on the DNA. So do all mutations result in a change in phenotype? Exactly, no. It depends on the environmental context. There's more about this later on in the presentation or in the other cram sessions.
Okay, thank you. Sorry, I forgot how far along you were going. All right, and again, mutations, I know if that's something that you're really struggling with, put it in the Q&A and ask a question, and it is something that we can revisit tomorrow night, because I do have some time in the presentation I've created for tomorrow, because any concept is fair game. And so if it's something that you want to talk about tomorrow, please just let us know. And if mutations is one of them, like, oh my gosh, I need more than one slide on mutations, just again, let us know. Let's move on to protein synthesis. So when I was a student, I remember learning about protein synthesis and I felt like I got it. And then all of a sudden someone said gene expression. And I was like, what's gene expression? And then I realized that gene expression is protein synthesis, right? If I'm making a protein, well, how did that happen? Well, a gene was expressed, right? Gene was transcribed and translated. And then I think a couple years later, someone said something about the central dogma. And yep, that was the same thing. Protein synthesis, DNA to RNA to protein. And so it's just one of those funny things in biology where they just sometimes have 5 different things that mean the exact same thing. I don't know why it is, but it's just something that needs to be said, because I remember as a student being like, what? And then once I stepped back and made sense of it, it made sense. But anyway, so as far as, knowing exactly what transcription and translation are, transcription is when we're going from DNA words to RNA words. And the reason why I'm saying words to words is that it's the same language, really, right? It's the ACTG, and then RNA is ACUG, right? The only difference is that there's no thymine, there's uracil. Translation is different because it's RNA words to amino acid language. It's not the same thing. It's not just a nucleotide anymore, right? It's not nucleotide to nucleotide. It's nucleotide to something completely different, a molecule called an amino acid, which is your building block for proteins. The exception to that rule, when we talk about transcription, is that some viruses with an RNA genome do this backwards because they have an RNA genome. Their genome is RNA. And so they actually go from RNA to DNA using an enzyme called reverse transcriptase. If that's something you've never heard before, totally okay. It's just an illustrative example that's used within this curriculum, but it's just something to be aware of. All right. I'm going to ask this again because this is something that I also struggled with as a student, where it seemed like every topic could come back to eukaryotic cells and prokaryotic cells. So as you're learning biology, just keep asking yourself, like, is it the same in eukaryotic and prokaryotic? Because they love asking that kind of stuff on this test because it's such a minute detail, but it's like really consistent throughout the year that like we're talking about living things, but eukaryotic cells are completely different than prokaryotic, right? Because they don't have a nucleus. So with that said, where does transcription take place and translation take place in a eukaryotic cell that has nucleus and a prokaryotic cell that doesn't? Take a moment, think about it. You don't have to answer, but you can if you want to. I'm going over so many FRQs tomorrow. Tomorrow is going to be FRQ City. All right, so I made a chart for you. It's also in the study guide. It'd be a place where you could fill this in. But eukaryotic versus prokaryotic. Prokarya is easy. They don't have a nucleus. So guess where it's all happening? In the cytoplasm, right? It's just, that's where it's all happening all at once. Whereas eukaryotic, we start transcription, or we do all of transcription in the nucleus, because if you think about it, like DNA is so precious and we have to protect it. And so it's in the nucleus. It's stored in a really safe place. When the RNA message is made, that can then leave and go into the cytoplasm and go to the ribosome. All right. You're all doing so well. I know this is hard stuff. DNA replication's big, DNA structure's big. Madison's going to take you through the beginning of transcription. So whenever you're ready, Madison.
Perfect. Great. So I'm going to go quick on transcription. So transcription, my apologies. Transcription mainly has three parts: initiation, elongation, and termination. So initiation begins where RNA polymerase unbinds the two DNA strands, so no longer being formed as a double helix. And this begins with the promoter. And the promoter is the DNA sequence that actually starts RNA polymerase in starting that pre-RNA strand. We move later on to elongation, where the pre-MNR strand will go to the opposite side of the template strand, and it will actually develop a longer pre-RNA strand of codons. And codons are the three bases of nucleotides that actually later on form an amino acid. And with that, it will continue producing until it hits the terminator, which is where we move on to the termination stage. And the terminator is the DNA sequence that actually stops the RNA polymerase from further transcribing the DNA strands. And therefore, at the end, polymerase will just stop working, and pre-RNA strand will then come out and go outside the nuclear envelope to further be changed, modified, and become polypeptide chains that will then become a protein. Moving forward, as we know that the pre-RNA strand is produced, there is many modifications that it has before it is actually sent out to become made into the other stages. So the pre-RNR strand is modified with the addition of the poly A tail added to the 3N, the GTP cap added to the 5N, and the introns are taken out and the exons are spliced. The exons are spliced because when the introns are taken out, the exons actually together actually make that production of the amino acid that is later on made in the other stage, which is called also called the termination stage, where oh my god, my apologies, which also happens in the elongation and termination stage outside of the nucleus, which actually makes those amino acids and therefore the protein. And I'm going to actually shift this over now to Ms. Kari, where she will answer the question of, does this happen in prokaryotic cells?
Thank you, Madison. All right. That was a lot of information, right? Transcription and post-transcriptional modifications. Man, you probably learned that in an entire week of school, okay? but we're just helping you through this. We've got this. So does this happen in prokaryotic cells? I put a poll up for you. If you could hop over to the little graph icon, it will bring you to the poll, and it's asking, does post-transcriptional modifications happen in prokaryotic cells? So take a moment and vote, please. So are all these things happening to a prokaryotic cell after transcription? Are they adding the poly A tail? Is there a GTP cap or introns taken out? All that stuff. Thank you for your participation. We've got about 80% of you saying no, and I would agree with you. This does not happen in prokaryotic cells. This is unique to eukaryotic cells where there's a lot of effort put into protecting the RNA. And here's why. The reason for putting a poly A tail of a bunch of A bases and putting this cap on those first two bullet points there, those addition to your RNA molecule is to protect it from degradation. And what I mean by degradation is breakdown, because once you're in the cytoplasm, that RNA is subject to breakdown from all sorts of things in the cytoplasm. It's not like it's taking a clean highway out to the ribosome, right? It has to travel in the cytoplasm and go to the ribosome and to be translated. So all of those things are there to protect it from cytoplasmic enzymes. Whereas someone in the chat mentioned how in a bacteria or prokaryotic cell, it's transcribing and translating like right at the same time, pretty much, right? So they don't need those modifications like a eukaryotic cell would. So really, really good job, everybody. All right, translation. Go ahead, Madison.
Great. And actually, guys, I am so sorry. I did talk really fast, so my sincere apologies on rambling a lot within the other two slides. However, I'm more than happy to explain a bit more of what I have talked about in the recent two slides in the chat, as well as the student host can also help out in the chat as well. So we move forward within translation, and the actual picture is showing the instance where when the pre-RNA strand is modified, it actually goes within ribose. So ribose goes on top of the pre-RNA strand. And with that, excuse me, the codons are translated, and therefore that produces many polypeptide-like acids. It makes many amino acids, which therefore becomes a polypeptide chain. And later on, the next slide kind of show you how it becomes a protein. However, once again, it occurs on ribosomes in the cytoplasm. And I'm going to ask you guys,
Oops.
oh, actually, sadly, I was actually going to ask, it's fine, no worries. I was going to ask you guys, where exactly does this occur on which ER, but it is the rough ER in eukaryotics. And yeah, within these, we have discussed the 3 steps, which is initiation, elongation, and termination. However, some defining features are that actually for this process to happen, similar to the similar discussion at the start of transcription, it starts with the promoter. So it starts at the start codon, which is also known as a promoter that initiates the start of these polypeptide chains being made. And once again, mRNA is read in triplets, which is known as a codon, which I've said before is the 3 nucleotide bases that actually produce amino acids. And tRNA brings the correct codon to the ribosome. And lastly, similar to what I said earlier in transcription, the process will continue further making that polypeptide chain until it has a stop codon, which is kind of similar to a terminator where it'll just stop that polypeptide chain from making. And the release factor, which is actually a specific protein, will go on and end that process from happening. So next, we'll show actually a codon chart, and codon charts are really helpful in actually knowing which amino acid is being produced per codon. So I will be asking you guys, looking at the chart, which amino acid would be brought to ribosome for the codon that has CUC? Yes, I have seen the chat and you guys are correct. It is this sign. And additionally, I'll be asking another quick question. Many amino acids are encoded by more than one codon, as you have seen here, which I was just getting discussed before. There's 3 nucleobases in a codon and therefore kind of produces the amino acid. Do you guys know why this is beneficial? And as you guys are thinking about that, I did not expect you guys to answer it like fully in the chat, but I will go over it now. It's because mistakes can happen and it does not guarantee that the amino acid sequence will be altered. And like I said before, the next slide kind of actually details on after translation. So after the polypeptide chain is made, which you see here, it is only made on its primary structure, which is just the chains of the amino acids. Then it folds to become later on, as you see, where it'll have become an alpha, oh my God, my apologies, an alpha helix, then a secondary structure, which is also known as the pleated sheet, then a tertiary structure, and lastly to a quaternary structure. This is the case of protein folding, where the peptide chain would just continue folding to become a protein. And if you guys have any questions, do not be afraid to comment. However, please do check before by looking at the Q&A tab to see if anyone else has asked a similar question, and be sure to upvote it if it has not been answered yet. And now I'll move it over again to Ms. Carey to let her discuss the regulation of gene expression.
Thank you. Can someone move me over? Wait, let me, I can figure it out. Oh, I figured it out. Took me an hour and a half. All right, so I'm just gonna pause for a moment. I'm gonna stop sharing my screen so we can have a little second to like calm down. Unit 6 is so hard, right? It is very hard. Transcription, translation, and all the microbiology. It's just hard. We can't see it. So what I want to help you with before we take a few minute break is something that I tell my students to do. And so if they're watching, they're going to be rolling their eyes at me, but I think they love it. When you're dealing with a difficult topic, I would draw out your current understanding of it, right? So instead of trying to look at a really complicated diagram, right? Like draw out, like I would draw for myself a DNA molecule and be like, all right, what do I know about transcription, right? And maybe I'd write like a pretend sequence and I'd say, okay, there's my gene. Transcription is just getting this into RNA language, right? And so maybe I would draw, okay, A becomes U, C becomes G, T becomes A, blah, 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 right? And maybe I'd label all this stuff, right? Maybe I'd make a little note saying that this is happening in the nucleus, okay, in a eukaryotic cell. So the diagrams are scary. The information is sometimes really scary, but again, like draw what you know and then go from there. Yeah, I see someone saying use fun colored pens, right? So doing all sorts of things like that. Yeah, so someone's asking a question about the coding versus the non-coding strand. I am going to make a note because I know that that's really difficult. Basically, the non-coding strand is the strand that RNA polymerase connects to. And I'll get a diagram made for tomorrow to have that make a little more sense. All right. So everyone go ahead and take like a 2 or 3 minute break. I'd even just like, don't look at the chat for a second. Just give yourself a 2 or 3 minute break, and then we will come back to our presentation. All right, I think that was about 2 or 3 minutes. So hopefully we were able to get something to drink and have a snack or go to the bathroom. And again, if you're feeling overwhelmed, just step away. It's totally okay. Again, I'm so just proud of you for being here and on a Saturday night and committing yourself to reviewing AP Bio, right? To do well on Wednesday. And again, it's just one test in your life and you're going to take it and you're going to answer questions, but it's not going to make or break your entire life's trajectory. Okay. But the fact that you're here is huge. And if there's any topic in particular where you are it's a major cause of stress for you, it's okay to let it go. You know what I mean? Like at this point, it's okay to know that it's not the majority of the test. It might be 2 questions. So give yourself permission to let that topic go and say, you know what, I tried my best. It's just stressing me out, and focus on something else. Okay. All right. You can tell I like to give pep talks. I bet it's pretty funny. All right, let's keep going here. Now, after translation, we've made this sequence of amino acids. And if you remember from unit 1 that Mrs. Shoemate covered yesterday, you should have learned about protein folding a little bit. Now, there's 4 levels of protein folding. You've got your primary structure, which is just your plain sequence of amino acids. And then just naturally, because they are molecules that have functional groups, like things with oxygen in them, things with nitrogen in them, they are going to start to be attracted to each other. It's just like I always tell people, well, not everybody. I don't tell people who are not in biology because they're like, what are you talking about, lady? If you put a bunch of people in a room, certain people are going to gravitate toward each other. It might be, 2 people know each other from a class they took, or it might be, hey, you're related to my best friend or something like that. And so naturally, people make connections and they'll kind of clump together in a room. And that's how protein folding is for me. You've got this strand of amino acids and they begin to interact with each other. And as they interact, they will eventually reach a 3D structure where then it is a functional protein. So not every protein reaches the quaternary structure, which is that 4th level. The quaternary structure is when you've got 2 or more polypeptides, which polypeptide is just a fancy term for a protein strand. Quaternary means you have more than one subunit that were made separately. So like hemoglobin is an example where you can't just have one hemoglobin. You need to have 4 subunits in order for it to be its functioning protein. So with protein, again, it's structure and function. So if it folds right, if the sequence is correct, if the environment is correct, then that protein is going to be able to function well. Oh no, it gave the answer. Sorry. So this question, I was going to put a poll up for you, but I gave the answer. So when we talk about regulation of gene expression, it's about, are the genes being expressed, right? And not all genes should be expressed all the time. And this actually reminds me of a question I saw in the chat when someone said, how do I know introns and exons? Well, exons are the portions that will be expressed. So they are the portions that don't get cut out because we want to express them. Okay, so exons need to be expressed, and that's how you should remember it. Introns are the ones not expressed, so we got to get them out. Hold on, that was weird. Okay, so no, this is why genes need to be regulated. I wrote there that the human somatic cell, somatic just means body cells, not gametes. We have 20,000 genes in every single cell. So if every cell was expressing every gene, it would be a disaster, okay? A structural cell does not necessarily need to make proteins for movement or transport. Okay, so this is where we get into like cell differentiation, like why a muscle cell is what it is compared to a bone cell. Well, they have different genes that were expressed and are continually different genes being expressed so that not everything is expressed all the time. That would be a big waste of resources. Another way that instead of just saying like, oh, genes are on or genes are off, Another way we can regulate gene expression is how our chromosomes are organized. So if you remember, like an actual chromosome is DNA that's super coiled into that shape of a chromosome. All right. They only do that when they start prophase. They're only going to like do that when they're about to do cell division. So another way that we can regulate gene expression is just by how DNA is organized. So in this picture, I have DNA, which is represented by like the purple rope, and they're wrapped around these proteins called histones. And you don't need to know that part, but I'm just trying to show you what this diagram is saying. And if you look at the top, it's got like a little thing that says gene, and it's pointing to a small segment there. Now, if that is coiled up, right, if that gene has all these proteins and other DNA, or the DNA kind of coiled up around it, it's not likely going to be expressed, right? RNA polymerase isn't getting in there, right? The promoter site, not going to be looked at. And that is one way that we can say, hey, you know what? This is a bone cell. All those genes for muscle cells, it's unnecessary. Let's just coil it up. We don't need it. Okay. And you might have learned about methylation or acetylation. And so I'll talk more about that tomorrow night. But basically, there are these chemical tags that can be put on the chromosomes, and it will either open up gene expression or close it down. And here's something really interesting that I like, it still blows my mind as an adult, that stuff that we do in our life right now. So drinking, drugs, like the illegal things that we try to prevent people from doing, they can change the methylation and acetylation of your DNA. So there are things that happen to you in your environment. That's why it's called epigenetic. Epi means like surface level. It's not changing your nucleotide sequence. It's changing the accessibility to genes that your body needs. And so if you're interested in that, there's like tons of articles all about it, all about like addiction and all sorts of things. Even like maybe if your teacher talked about like the mouse pups who were either licked by a nurturing mom or not, and how like if they were not nurtured as a baby, they then had children who like couldn't handle stressful situations because it altered their DNA, which just blows my mind. Another thing that can happen with regulation of gene expression is having regulatory sequences and regulatory proteins. So if something needs to be expressed, a eukaryotic cell would have all these different transcription factors. You might have learned about those transcription factors. They're basically proteins that help recruit and allow for easier transcription. So you can see in this image, there's all these little circles that are different colors. Those are mediator proteins or transcription factors. But then on the DNA itself, there's that promoter sequence and there's enhancer sequences that are going to be downstream, which means just like on the other side of the promoter of your gene. So there's a lot of complex stuff going on in order for one thing to happen, in order for gene expression to happen. And so we could get into a lot more detail with this, but we don't need to right now. I would pay attention to if you see the word regulatory sequence versus regulatory protein, that'd be a really good reading strategy of, hey, if they're asking you about a diagram, if it's a sequence, it's going to be on the DNA, right? It's a sequence, just like a DNA is a sequence. It's a part of that whole thing. And then proteins are going to be things that are helping recruit RNA polymerase or helping shape the DNA into the spot it needs to be for transcription to occur. Okay, so I hope you're feeling okay. That was a lot of information. We talk about regulation of gene expression, but again, just always take a step back and be like, why are they regulating gene expression? Oh yeah, okay, because we don't want to make it all the time, or something is changing and we need to make more of something, right? And this person's in a stressful situation, so let's release some cortisol, right? Let's get things moving. Prokaryotic regulation. I saw some people talk about operons. They were like, lac operon. Oh my gosh. So we've got prokaryotic cells like a bacteria, and they regulate their gene expression in a different way. They have these things called operons, which basically means you've got several genes being controlled by one regulatory system. All right. So this diagram's big, but if you just take a look at it, in the very beginning, we've got this DNA, RNA, it's making this purple donut-shaped repressor protein. And if you see, this repressor protein is on the strand by the letter O. So it's by the operator sequence. Oh, thank you for that feedback that they can zoom into the presentation. I love that. Yeah, this is as big as I can make it for you, unfortunately. So yes, please go ahead and do that. So anyway, so an operon, it's a cluster of co-regulated genes. They can either be inducible or repressible. So let's do a little poll. Is it? I'm making the poll right now. Stand with me. Is it inducible or is it repressible? So this image is showing that when lactose is present, lactose is binding to the repressor, changing the shape of the repressor. And then RNA polymerase can come in and do its thing and make those proteins. Okay. Great. All right. You are at like 98% saying inducible. So we know operons well. I'm very, very happy about that. Great job. Yes, it is inducible, which means it's normally off, right? Normally, if you're a bacteria and there's not lactose in your environment, why would you need to be making enzymes to digest lactose? That seems like a really big waste of space and materials. But if lactose is in the environment, then you're going to want to make those enzymes so you can actually digest them. When lactose is present, it is binding to that repressor protein. The repressor protein has a conformational change, which means it changes its shape. It pops off so that your RNA polymerase can come in, transcribe, and then eventually we get the translation to make those enzymes, right? I added in some vocab here. Again, you have access to these slides if you click the green button at the bottom. And so if that's something you really want to like put your head around again, then that's great. And let's sidestep and do some biotechnology. If your teacher did not get to the pGLOW lab or a similar lab where you're looking at genetic transformation, genetic transformation is an example of how basically we can insert a gene of interest into a bacterial cell and it will start to express that gene and make the protein that we want it to make. So for example, in the real world, we can put a human insulin gene into a bacteria and then that bacteria can make boatloads of insulin for us and we can put it into vials and give it to people who need it. But the pGLOW lab is a fun version of that where basically you took a plasmid that had the jellyfish gene. It also had a antibiotic resistance gene. And we'll talk more about this tomorrow because I do want to dig into it with you. But then we put it into a bacteria. You did the whole heat shock thing. The bacteria took it up. And then 24 hours later, it hopefully glowed because that means the bacteria was transformed. The bacteria had new genes and the gene was being expressed and making that glowing protein. Some other biotech that you'll be responsible for knowing, one process is called gel electrophoresis. So electro, meaning there's electricity involved. The phoresis has to do with like the pores in which the DNA travels through. It's usually in the context of this course talked about in terms of DNA being separated, but you can separate protein as well. But likely you did a lab or maybe a virtual lab where you had strands of DNA that were then put into these wells and they traveled toward the positive end because DNA is a negative molecule. And it helps us just separate things according to size and charge. So if we want to figure out how big the DNA is, or if we're trying to figure out, hey, whose crime DNA, whose DNA was left at the crime scene, we can compare it to suspects. PCR is another technique called polymerase chain reaction. And that means we are amplifying DNA, which doesn't mean make it bigger. It means make more copies. So your teacher takes one copy of something to the printer and makes 150 for all the students. Okay, so PCR is like a copy machine. DNA sequencing is another thing that you didn't necessarily have to do in class, but it is good to know. And it is what it says it is, DNA sequencing. We're looking at the actual order of nucleotides. There's usually, computers that do this, so you don't need to do it by hand. Okay, but as far as biotech goes, that's about all you need to know. Again, it'll be more application questions and things like that. All right, so we are on practice questions. So wait. I'm going to share a new screen with you. So get your phone ready to take a QR code. And I'm going to get the quiz up for all of you. So one moment. Ta-da. I'm going to share something new. Hold on. I have to hit the button. Hold on. Sorry, everybody. Just take a moment while I figure this out. Okay, I think I got it. Here it is. All right. The QR code will come up in just a minute for you to join our Unit 6 practice questions. These are not going to be as hard as they are on the exam. So take a picture of that QR code so that you're able to join us. Again, these are just short multiple choice questions. They are not as difficult as the ones that are on the AP test, but it's a good practice for all of us. All right, I'm just going to hit start. We have a lot of people joined. And actually, you know what? I'll also put it in the chat. So if you don't have a QR code situation, there you go. Let's get started. First questions about DNA replication. You got this. All right, let's see this answer. Perfect. So DNA replication is semi-conservative because one strand serves as a template for others. Great job, everybody. All right, good. So instead of having thymine in RNA, we have uracil. Excellent. Amazing. 91% DNA, RNA, protein. Let's do a couple more. If I hit the next one, let's go. Good job, helicase. Excellent. Let's do one more. Beautiful. Topoisomerase takes care of that messy supercoiling. So good job. I want you to know that for all of these quizzes, I do have more than 5 for us to take, but I am only doing 5 questions at a time right now so that we can make sure we get through all of our units. So if there's time at the end this evening, we will go ahead and open those quizzes back up and you can take them and we will talk about the answers. So really, really nice job. All right, how are we doing? Do we need a break? I'll make a little poll. Let me make a poll real quick. All right, please answer the poll. So I get a sense of if we need a break. That's perfect. Okay, I don't know. Why did I get... Can someone fix my screen for me? I don't know what happened. It's like all of a sudden I'm half the size. Thank you. Okay, 75% of you want a break. I hear you. I hear you. So I'm gonna go ahead and do this 1.5-minute timer. I might set it twice so we get a good 3 minutes in. It's not gonna be a loud alarm clock. It's gonna be the sound of crickets at the end. Okay, so go ahead, go to the bathroom, do whatever you need to do. I'm also gonna go take a quick break. So again, it'll be the sound of crickets. I'm setting the timer one more time, everybody. righty. Those are the crickets calling you back from your break. We just have a little over an hour to hit our last 2 units. We are doing really great in terms of timing. Give me a second. Let's pause that. I don't want that there. Okay, our next unit is Unit 7, Natural Selection. So Madison's going to hop on and kind of guide you through a few slides, and then I'll come back after that.
Great. First of all, you guys are halfway there. I am so proud of you guys being able to crush it these past units, and I'm just honestly so proud of you guys continuing throughout this cram session. I'm honestly just very proud, and all of us are very proud of you guys for moving forward. So we'll be now discussing our 3rd unit on natural selection and evolution, which has 13% to 20% of your exam. Next slide kind of shows you the key vocabulary. My apologies. The key vocabulary that you guys should know that we also recommend for this unit. However, if there is terms that you do not understand or unsure of, the Cram Hub is a great way to maybe be able to figure out some of these terms, as well as the resources Fiveable provides as well. And at a glance, this shows you guys the concepts that are discussed in this unit. And in the middle, you guys do see that there's 2 specific skills. And if you guys are unsure and confused of these skills, we will be showing examples of both skills and will be addressed on nights 3 and 4. So do not fret. Now I'll give it over to Ms. Curry.
Thank you. All right, so like Madison said, this unit is a larger portion of the exam, but in my opinion, I think it's one of the easier units to do well on. A lot of the things that Charles Darwin figured out and current evolutionary biologists, it makes sense, right? Like the evidence points to a conclusion that the average person would say, oh yeah, that does make sense. So the big things that I'm going to be going over tonight are natural selection, just because it's the baseline, right? And so if we don't have the fundamentals, if you think about it in sports, right? If you're all of a sudden just going to jump in, you really need to know the fundamentals. So that's the fundamental of evolution. And then I'm also going to really hit phylogeny, because evolutionary trees are on the exam, like most of the time. Watch the 1 year that I'm telling everyone, yes, let's know trees. It's not on the exam. I don't know. But it's a really good skill to have. It's a really good understanding to have. So let's make sure we talk about phylogeny. Now, when we think about evolution, there's micro and there's macro. Micro meaning small, and micro is a change in the allele frequency. So and that's why we learn genetics first, right? Before you learn evolution, so that you can have that genetic understanding of alleles, right? Like if you have a population of beetles, and the most prevalent allele gives them this really bright green color, and all of a sudden the environment changes, right? All of a sudden that large proportion of green alleles is at a disadvantage. And so maybe over time we'd see that change over time because the environment changed. Someone's asking about Hardy-Weinberg, and we will get to Hardy-Weinberg on the math night. So not tomorrow, but the next night is going to be all about math and graphing and things like that. So thank you for asking that. Now, how do allele frequencies change? First, I kind of mentioned natural selection. Okay, so if the environment's different, those who are most fit to the environment are going to most likely reproduce and pass on those genes. But also changes in allele frequencies can happen by mutations, right? Just by simple mutation. And all of a sudden, if your teacher taught you or had you go through that rock pocket mouse thing, okay? If you did rock pocket mouse, you probably watched a little bit of the video and how we've got like the lighter colored mice who were living on the sandy area, and then the black rock from the volcano had darker colored mice doing well. And so that actually ended up being from a mutation. We've also got gene flow, so just genes moving in and out of an area, and then genetic drift, which 2 examples of genetic drift you would have seen are the bottleneck effect and the founder effect. So the bottleneck effect is generally when there's some sort of external like tragedy, right? Like there's a tsunami or there's this wildfire, and so only a small portion of the population survive, and then they are the new population. Founder effect is that picture here with like the butterflies, where you have a small population who leave an area and they go found. They're like the founding animals of that organization or of that new area. And then non-random mating, I love this little GIF there where it's the sexual selection piece. And so a change in allele frequency can happen when you've got non-random mating. It's organisms that aren't just like, hey, randomly, these two organisms are going to make offspring. Especially with birds, you might have seen really cool videos of like the birds in paradise where they look for a certain dance or a certain brightly colored feather situation. And so those would be causes of frequency. I see a lot of people talking about FRQs. Tomorrow night is going to be just filled with FRQs. Filled with FRQs, I promise. In practice tests, you can ask your teacher to open up questions on your AP classroom. And so that would be a really, really good thing. So someone's asking about gene flow, genetic drift. So gene flow is like immigration, emigration. It's just the flow of genes in and out of an area. And genetic drift is something a little bit more major, like a catastrophic event environmentally where a small population are the sole survivors. And so the frequencies change because all of a sudden we had, 2,000 beetles and now we have 10, and they are the founding organ, if they've moved somewhere, they're like the founding alleles for that population. All right. Natural selection, again, is Darwin's theory. It is supported by multiple lines of evidence. And I'm sure you learned this, but he was all over the place and just making tons of drawings and observations about specifically birds and animals and how they just seemed to be so perfectly suited to where they lived, and he was just couldn't believe it. And the most famous example is all these different finches. And his hypothesis was maybe there was an ancestral finch that was just one type of finch. And maybe the different areas of the island that had different types of seeds, had different types of habitats that were quite different, allowed for evolution to happen. And so just by random variation that already existed, we can eventually, over a period of time, have a variety of species. I'll give you a minute to look over the principles of natural selection. If you can't read it, I apologize. But even if you just look at the 4 main titles, go ahead and do that. I gotta say, I love that I see all of you helping each other in the chat, answering questions. I really, really appreciate that. So natural selection has to do with environmental pressures, right? So I'll explain the picture, sure. So in a population, which a population is organisms that are of the same species living in the same area at the same time. Within a given population, there's going to be genetic variation. We know that because of meiosis, right? And from sexual reproduction, there's genetic variation because of that. So in this picture, it's showing a variety of insects of sorts. There's some blue, some green, some brown. And right now, with the ground being brown, the brown organisms have a better chance of surviving. And so the first part was variation. The second part is overproduction, which basically just means there's not enough resources for every single individual to survive. It's just a common sense thing of like, there's something that gives up. And maybe it's room for shelter. Maybe it's resources. Maybe it's finding a mate, right? It's not possible for all to survive. So because of that, selection takes place. And so in this case, it's a bird that can easily see the green and blue insects. Okay, so sometimes selection is from a predator, but sometimes selection happens because of competition, right? So competition for food or resources or mates. So it's not always about getting eaten, it's about just surviving. And sometimes sometimes it's the lack of food or the lack of shelter to survive, or it's the lack of having a trait that is okay with the environment. If all of a sudden there's a sudden drought and you're a plant that just so happens to be really susceptible to drought, then it's unlikely that your genes will continue on. Whereas if your variation happens to be, hey, I'm a plant that can do really well, then you'll survive in that drought a little bit better. So simply put, nature selects which organisms are more likely to survive and reproduce. That last part of surviving and reproducing is the key to natural selection. So it's not just about like, hey, who wins, who's better at their environment. It's because you can survive, you are more likely to reproduce. And if you reproduce, your genes that helped you reproduce are now passed on. The other key thing, do not say that an individual adapted. Okay, adapt is not a verb. We're not going to use that. A population can change over time. And especially like in an FRQ, you wouldn't want to say, oh, population will undergo natural selection. You're going to want to break it down and say, hey, the environment did this. So this population, this portion of the population with this trait are more likely to survive. And because they're more likely to survive, they're more likely to reproduce, which means their genes are passed on, which might become more prevalent. All right, another key thing in evolution is this common ancestry theory. Okay, so when we think about common ancestry, I just drew this tree down. It's a really big simplification of all living things. But basically, all living things fit into these three categories, either bacteria, archaea, or eukarya. If you've never heard about archaeobacteria, don't worry about it. I'm just showing you this to show that like all living things are a part of this tree. And all living things have these three things. They all have ribosomes, they all have DNA, and they all have some sort of outer membrane, right? Even if it's a single cell, they all have a membrane, something that's protecting them from the outside. So RNA is a really good piece of evidence to say like, wow, or ribosomes and DNA are really good evidence for, wow, all of these living things are connected. Another big thing is the unification of eukaryotic cells, how all eukaryotic cells have several very similar structures like linear chromosomes, genes that have introns, and membrane-bound organelles. So membrane-bound organelles are just organelles that have their own membrane, right? Like a mitochondria, a lysosome, like endoplasmic reticulum, right? They all have a membrane so that they can compartmentalize and do their job. So these are just pieces of evidence. There's also features and processes that are conserved. So if you think of something like glycolysis, like every living thing does glycolysis to get ATP, to make ATP. So when we talk about the word conserved, I want you to talk about that in the chat. What does it mean when a question or a paragraph of information says that like, oh, this trait has been conserved over time, or this process has been highly conserved? What does that mean? Yes, I love this. Some of you are saying it's maintained over generations, kept through generations. Yeah. Good. Good. Yeah. So conserved, and not just generations, but it's maintained, it's stayed relatively stable, right? The process itself, the thing itself has not changed much over millions of years. And so conserved, think about it like that. It's not just like from one generation to the next. It's like everything. It has this similar process or similar trait. All right, I'm going to jump into evidence of evolution for you all. So there's three main buckets when we talk about evidence of like, how do we know that things are changing over time? Well, at first we looked at fossils, right? We still do. But we're looking at layers and we're saying, hey, look, there's a similar thing from here and here, and it looks similar in the next level, but it's changed a little. We can also look at radiometric dating. And so they look at the rate of decay of carbon-14 is just an example. So they look at, You don't have to be able to calculate any of this, but carbon-14 is an isotope of carbon. And so there is a known decay rate. And so based on how much of the carbon is decayed, they can figure out mathematically, like if you take AP physics, I think you eventually get into that, they can tell you how old something is. Biochemical is where we're comparing DNA or RNA or protein. And so that can help indicate common ancestry and relatedness because, hey, if this whole sequence is nearly identical, that tells us something. That tells us that they're probably related or probably pretty closely related. And then another one is morphological. So that would be like your homologous structures you probably learned about, the vestigial structures, so those structures that don't have a function anymore, and analogous. Homologous structures are like the ones pictured. And that's why I put that picture there. So homologous means, wow, even though they don't necessarily look the same, they function very similarly. So for example, like in this picture, I know it's hard to see, but it's like each of those organisms has a long bone and then a segment with 2 bones and then a segment with small bones and then a segment with digits. Okay. So they have this same blueprint that it might function differently, right? Like if it's like the wing of a bat or something like that. I think I said it wrong. I think I said function the same. That was my bad. So the structure is similar. The function might be different. But the key thing for you to know here is not the definition. The key thing to know is that homologous structures can serve as evidence for evolution. Okay, or as common ancestry. All right. Some other evidence that evolution is ongoing, that it's not just like something that happened in the past that we dig up in some rocks, but we get new pathogens, right? Like COVID-19, right? There was changing pathogen. We also have antibiotic resistance. So we have these bacteria who are changing and they are being exposed to different things in nature, and then certain ones survive because they're more likely to, because they have resistance to it. We also have just changes in the genome over time. And then we can also just look at continuous change in the fossil record of things changing. And so that are all good evidence that evolution is ongoing. I'm going to jump into speciation. Speciation is when we are getting a new species. So formation of a new species. We should probably talk about what is a species, right? So we use the biological species concept most commonly accepted within science. Basically, a group whose population can interbreed in nature, they can produce viable, which means living, fertile offspring. Fertile meaning their offspring can have offspring. And then they do not interbreed with other populations. So that kind of keeps them separate from one another. You know, if you talk about, your teacher might have talked about like the liger, how like a lion and a tiger can mate, but they're still 2 distinct species. But that's because their offspring, the liger, is infertile or sterile. Yes, good job, everybody. So yeah, so that would be kind of why lions and tigers are an example of they're still 2 different species, even though they can interbreed. And yeah, mules, good job. Yep. So things like that. When we think about how that happens, how does a species even form, right? Like, why don't we just have all the species we're ever going to have? Why can we have the possibility of new species? So how does this happen? Gene flow has to stop. So a population has to be isolated, either geographically, reproductively, temporally, so in terms of time, behaviorally. There's some sort of isolation that happens for long enough that breeding between other populations don't happen. And then just by natural changes in the genome over time, like mutations, even if they're small mutations that don't do anything, long enough time might happen where then they can no longer interbreed. All right, good. Okay, so how do we prevent gene flow? So one of the classic examples is a geographic isolation, right? So this picture here is showing some mountain ranges. The original population covered both mountain ranges. For whatever reason, they're either geographically separated, like one population is at the peak of one mountain and the other is at the peak of the other. So they are geographically separated by 2 different mountains. And over enough time, they'll experience a change in their nucleotide sequence, which then maybe is changing in their traits. And then if we were to bring them back together, they would be reproductively isolated. And I'll talk about reproductive isolation in a minute. This picture, I know it's really small, but this is the sample of separating this population of flies and giving them different food sources and eventually trying to put them back together. But now they have kind of like a behavioral separation where then they like a different food source. And so potentially would become two new species, but that's like a lab example, not necessarily from what's happening right now. Sympatric speciation means it's happening like right next to each other. It's not happening allopatrically. Allopatric means it's happening away from each other. So allopatric, A for away. They're away from each other. So sympatric speciation means like, hey, these two populations are right alongside each other, but they still somehow became separate species. So this example here shows like there's different times of day or different times of the year where two things will be ready to create offspring. Like there are these flies that prefer two different types of fruit, and they are the same species, but they both prefer different fruit trees, and those two trees ripen at different times. And so they're actually going to be not even near the other population when they're ready to mate because they're interested in different food source. So, sorry. Okay, I'm jumping into evolutionary relationships. I saw somebody asked about like the difference between phylogeny and cladogram. Technically, yes, it's written in the AP curriculum that you should know the difference. So phylogeny and cladogram, they both look at evolutionary relationships. Phylogenetic trees can talk about population over time. So they might have some sort of like x-axis where it says like 50 million years ago, 5 million years ago, whatever. But I've never seen it like explicitly tested before. And the goal is not to trip you up. So it's fine if you think of them as the same, but there is that little tiny distinction. But either way, they're showing relationships. Your outgroup is going to be the one. Oh, I'm not going to tell you that. Oh, never mind. It's already there. So the outgroup is the one that's kind of like on the outside, right? The least related to everyone else. So I always start by just trying to identify the outgroup, which one is least related. The ones that share a node are going to be more closely related. So let me see if I can pull that up for you. Here we go. Oh, let's see if I can do this. I think I can. Okay. Hopefully you see my laser pointer. So these little points here are your nodes. Right here is a node. Right here is a node. Okay. And at these points, this node right here, A and B could be on opposite sides, and it does not change the phylogeny. Right? They're still coming from that same separation, that same divergence right here. Also, these boxy looking ones, it's showing the same thing, but just some people prefer one or the other. So to construct trees, how do you do it? Well, long ago, they just used physical stuff. They said, oh, look, all these organisms have a backbone. Some of these organisms have lungs. Only a few have amniotic eggs. Only a few have this, and only one has that. And then they put them on trees to say like, hey, look at this evidence we have that, they're similar. Most reliable data is going to come from DNA and amino acid sequences, right? Because that's like in the cell. We're at the cellular level and it's coming from there. So I'm going to give you a second. Ooh, I got to make a poll. So look at that diagram and tell me which one is the out group. Hold on, I'm going to make a poll. Hold on. All right. So looking at that trait characteristic chart, which organism is the outgroup? Amazing. You're all so good. It's great. Wonderful. So a lot of you said, be the fish, right? So I would draw my tree with the fish way off to the left, and I'd say, you're the outgroup. And then I'd draw my other line to the right, and then just kind of keep working with, okay, who's the next least related, right? So like looking at this chart, I would say, wow, well, look at the frog. This one has only 2 shared characteristics with everyone. So I'd put the frog next, and then so on and so forth. Excellent job. All right. So this is an FRQ that we're going to work through tomorrow, but I wanted to show it to you. This is from 2015. They even give the template for the tree, which they've done that the last few years. So for those of you who kind of are really worried about just drawing a tree out of nowhere, they have been giving the template. That doesn't guarantee that they'll continue to, but that makes me really happy, because then it's not getting at, do you know how to actually draw the lines? It's, do you know how to place the organisms? So this one, I want you to just take a look at here, how it's talking about the number of amino acid differences. So we're not looking at similarities, we're looking at differences. So let's find the outgroup. This one looks funky because it's got all these weird names. And we're like, oh, shoot. So 0 and 0 means there's no differences. So E. ferris and E. ferris, that makes sense because they're the same species. We've got E. ferris and D. polylepis. That's 21 differences. So keep looking and try to figure out who's the outgroup. Who's the one that's most different? Make sure you're looking horizontally as well. Don't just look straight up and down. Okay. This would be an FRQ where you would have 10 minutes to figure it out. And I know a lot of you are saying this one, E. africanus, because you probably you were looking vertically and you saw the 20 and you're like, ooh, 20, 10, and 12, those are all pretty big differences. And I agree, those are. However, if we look at like 10 and 12, that's not like, E. africanus isn't that different from G. gallus and A. forsteri. That's only 10 and 12. But if I look to my left, it's D. polylepis is 17 differences with A. forsteri, 18 differences with G. gallus, and 21 differences with D. polylepis, or with E. ferris. So it's actually this one that's the outgroup. So I don't want you to get discouraged. This is why I'm showing this to you. So when you see something and you're like, oh, 20, yes, that's got to be it. Look at the whole picture, right? Let the evidence speak for itself. Make sure that all the evidence is supporting your first claim that E. africanus is. Again, we're going to solve that tomorrow night. Here's another one. This is saying divergence in percent of mitochondrial DNA sequences. So again, we're looking at mitochondria DNA sequences. This is going to be DNA from their mothers. And they give you a really nice tree and they want you to construct your diagram. And it actually even gives you the outgroup here. So for those of you who were like, oh my gosh, if I can't even find an outgroup, then how am I going to do this? But this one, they gave you an outgroup, right? Because that's not what they're worried about. They're worried about the rest of it. And can you continue to solve it? So tomorrow night, we will work on these. And actually, if we have time at the end of today, we'll just hop back and look at them. I see someone's asking a question about like, why would there be shaded in boxes? Is that you don't have to compare each pair twice. So for example, like human and gorilla are 10.3 differences. So in the other human and gorilla box, you wouldn't have to put 10.3. That would just be really confusing. So it's doing it that way. So you're only comparing each pair once. Yep. The shaded boxes are just a repeat. Okay. All right. You can all do this. All right, we got 40 minutes left. Someone's telling me. I'm kicking it. I'm going. All right, here's another example of like an application question. They might just give you the tree, right? And they're saying, hey, look, we've got all these different types of photosynthetic organisms, flowering plants, gymnosperms, which is just a fancy term for like conifers or like pine trees. You've got ferns, you've got mosses, blah, 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 all these other things. And down here, I just wanted you to look at, so hopefully you're looking at your screen and can see my little red thing. We've got these traits that happen, okay? So here's your common ancestor at the way bottom. Now, as things change over time, this embryo protection trait comes about. Everybody after this also has embryo protection. So even though it's only listed there once, everything above that, so from liver where it's on, are going to have that. Same thing with apical growth, okay? Apical growth, here we go. It doesn't matter. Do you know what apical growth is? Doesn't matter. But you can read this and say, yeah, I know which ones have apical growth. It's everything after that. So I'm going to ask you now, which organisms have vascular tissue? So based on what I was just telling you, here's vascular tissue. What are the names of the organisms that have vascular tissue? Or you can just give me a general answer. You don't have to say all the names. Yeah, love it. I love how you're answering it. Yay. Good job. Yes, so I'm hearing this. Everything after that, right? So club mosses all the way to flowering plants, they're going to have that same trait. And this is just another application thing where they might say like, hey, which organisms have this trait? Or, hey, this trait happened here. Where would you place it? Place it right there, right? Right. I'm going to quickly go over origin of life, and then we're going to move on to ecology. So unit 8 is where we're headed. So origin of life, you need to be able to describe the scientific evidence that provides the support for models of the origin of life, because we don't know like for sure how the origin of life happened. We have a lot of experiments and a lot of evidence. So here's a good timeline that you should be aware of, right? That the Earth formed about 4.6 billion years ago. The earliest fossil wasn't found, or isn't when they dated it to see how old it was. It was 3.5 billion years old. Oh, I have a typo, sorry. The Earth was too hostile before 3.9 billion years ago. So before that, that was like no way that life could form. So it was life began between 3.4 to 3.9 billion years ago. And it didn't just start with animals and plants, right? It was small organisms. There wasn't even enough oxygen. So thankful to photosynthetic organisms, oxygen became more abundant in the atmosphere. And so what the models are for how they are determining how life started, the idea is that there were these inorganic precursors that assembled into organic molecules. So organic meaning like, your carbon-based molecules, like your, kind of the precursors of your organic macromolecules. And then the other ideas, another model is that organic molecules could have been brought here by a meteorite. So the scientific evidence is written there. Experiments have shown that it's possible to form organic molecules with similar components present on early Earth. You might have learned about the Miller-Urey experiment, which is pretty famous. But basically what they did was they tried to simulate what early life was like, right? There was lightning, is the energy source here. They've got some gases from the primitive atmosphere. So some NH3, H2, water. And they were able to show that, hey, look, these small organic molecules were made from this experiment. And so that's just some evidence on some ideas of how this happened. Okay, that is it for evolution. Again, we didn't cover everything, right? Like we just couldn't. So I am going to give everybody another quick break. Or actually, no, just kidding. I'm going to do practice questions first. Let's do some practice questions and then we'll do a break. So I am going to go to the Unit 7 practice questions, and I'm going to be a better host, and I'm going to put it in the chat instead of a QR code. All right, so if that chat works, hopefully you'll get to your practice. I will start it. I know I'm tired too. We can do this. We're almost done. We're almost done, Nadia. All right, I'm going to stand up while you all take your quiz. For time purposes, I'm just going to reveal the answer. Thank you for those of you who Answered. Okay. If I had to summarize evolution in one sentence, what would be the best way to explain it? And D is correct. Yeah. Populations of organisms changing over time. The reason A is not correct, we don't say one adapt. Remember how I said adapt isn't really a verb? An adaptation is a thing, is a structure or a trait that is genetically derived that becomes more abundant and serves a purpose. But really be careful about talking about one thing adapting. It's not like, oh, we have to adapt because all of a sudden this change happened, right? Like that it is a population changing over time. Really good job. Let's do a couple more. Awesome participation. Wonderful behavioral isolation. Absolutely. Different courtship rituals and dances, unlikely to produce offspring. Very, very good. They're isolated behaviorally. Love it. Next question. Mine has weird wording, so oh, you are also smart. Great job. Yeah, and A was written to kind of mess with you a little bit. Like it just gave a little too much information. So when you're taking the test on Wednesday, read the answer option you choose carefully because sometimes the way these things are written have about half of the answer correct, and then there's something in the answer wrong. And so just double check when you are answering a multiple choice question. Just really look at the one you chose and be like, is this full? Like, did I really do this? Is that fully true? No, you do not need to know about nerves and physiology anymore. So the reason it's not A is that it went too far saying and form new structures. So fitness doesn't have to do with forming new structures. New structures can, right? Like adaptations can come because of natural selection, but it's not like a guarantee. It's not a principle of natural selection that, oh, if you survive and reproduce, then you get an adaptation, right? You don't get a new structure. So A just has too much information that's not always true. And that's why that's not right. All right, let's do two more. We got another unit to do. What 94%. I love it. Okay, so plants contain a variety of pigments capable of absorbing light. Why is this biologically beneficial? D is the answer. Different pigments absorbing varying lengths give plants a broader range for light absorption. Kind of goes along with like why genetic diversity is good too. It's like having a multiple set of options for survival is always good. All right, let's do one more. Good. All of these. Yep. So differential reproductive success just means some of the individuals in the population are more successful with reproduction, right? They're more likely to reproduce. Struggle to survive competition. That's that not everybody can survive, right? Not every single organism survive. And then genetic variation has to exist. If everything is a clone of each other, then there is going to be no nature selecting anything because everything's going to be able to survive and reproduce. So excellent job. I love it. I'm going to get my screen back up and we will finish this out. We are doing so well. We have one more unit. And again, we're doing 30 minutes per unit. Oh, all right. I'm going to, well, sorry. Hold on. Ta-da. All right, I am going to stop talking for a minute and let Madison take over for you. We're going to skip our break, okay? Everybody, we can do it. I believe in you.
Great. Before I go really quickly, congratulations for crushing the 3rd unit. You guys got this. Keep it going for our last unit. So our last unit is on ecology. So the next slide kind of shows you guys the key vocabulary of the unit. And remember that these terms will be mostly placed on like application-based questions. So you got this. Do not fret about if you may not know some of these terms. And looking at the unit at a glance, there will be the concepts. Oh, sorry.
Yep, I'm having technical difficulties. I'll get it going for you in just a second. It's loading.
No worries.
Okay.
Great. I just showed you guys the vocabulary. Also, just a reminder once again, there will be mostly an application-based question, so do not fret if there's, once again, some of the terms you guys do not understand as much. The next slide is just showing a unit at a glance. The highlighted words are the big topics we'll be mainly covering throughout this unit primarily. And on the side, you also see the topics that will be discussed as well. And now I'll give it to Ms. Kari for her to go over this unit.
Thanks, Madison. Okay, so I need my techie friends to help me. There we go. You're the best, everyone. Let's just talk about what ecology is, because I know that this is a unit that a lot of teachers in schools just kind of run out of time for, or you self-study it because it is an easier unit in most perspectives, right? It's not as like blow your mind microbiology, that's cellular respiration and transcription, translation. Like those are heavy units, and these units are like, oh, plants and animals and organisms, and I get it. Like it's not too stressful. So I love that we're ending on this unit. I love ecology. I actually teach it first semester because we live in Michigan where it's cold, and so if we waited until March to teach it, it would still be snowing here. And so we wouldn't be able to go outside and talk about abiotic and biotic factors. So I actually put this picture in here. This is my 3-year-old, and can anyone tell me what he's holding? Can you tell? And the highlighted topics, by the way, are the ones that I would be specifically going over tonight, not necessarily the most important. Anybody see what my son is holding in this picture? He's holding a snake. Yeah, it's a snake. I thought it was a stick too at first, actually. He spends a ton of time out in the forest in Michigan. He didn't pick up the snake himself, but my father-in-law did. And if you zoomed in on the picture, you would see a big lump in the snake's tummy. So it had a mouse, a couple hours before my son picked it up. Yep, it's a garter snake. Yep. Now, the reason I wanted to show you that picture, one, because my kid is super cute, but he's an organism, right? He's an animal, and he's in an environment. There's leaves out there. There's water. There's snakes that he could pick up. The mouse is dead. It's in the snake's tummy. And they're all existing around each other and influencing each other in one way or not, right? Like every moment that we're on this earth, we're affecting the environment, and the environment is affecting us too. And so that's what ecology is. Ecologists just look at stuff and they're like, I wonder what would happen if this happened, or I wonder why these two things are happening. Okay. Abiotic factors are the ones that are not living, like weather, right? Or water quality or water usage and things like that. Biotic factors, think biobiotic. They're the factors that are living. So it's like, hey, the predators in your area or the parasites that are trying to attack you. Your access to food availability are going to be all of your abiotic factors. So in the chat, what are some things that are part of you and your environment? What are some things that are around you that influence, your world? So think about it. What are some abiotic, biotic things in your life? Yes? I say snow. Access to water. Yep. I live on a little peninsula. I'm surrounded by tons of water. Music. Yeah. Your phone. Those are all abiotic things. Grass. Hopefully you eat, right? You have access to food. Awesome. Thanks for sharing. Other things we want to think about when we think about ecological frameworks. We can look at individuals. Like we can just study an individual organism and how it interacts with its environment. We can also zoom out to the population. So I love elephants, if you can't tell, which is why these are all here. Well, elephant population would be same species, same place, same time. So we could study a specific individual population. Okay. Community is zooming out a little more. We're looking at, hey, there's elephants interacting with zebras and interacting with all sorts of different populations of organisms. So we're zooming out a little more. The final thing we zoom out, at least for this class, is ecosystem. And so ecosystem is where that abiotic factor comes in. Okay, so abiotic factor does not come in until your ecosystem level. Okay, moving on. Oh no, I'm not going to have you type it in the chat, even though it says to, just because we don't have a lot of time. But you think about like, how do you respond to your environment? Like if you're in a classroom and the lights are kind of dim and somebody comes in and turns on all the lights, right? What happens to your body? Or if out of nowhere you get startled, right? Like someone jumps out to scare you, right? Your body reacts, right? Something changes. And so organisms respond to their environment too, based on different cues and different mechanisms. So like if you're a plant, right? You will bend, you'll respond physiologically to bend toward that light. Responses can be behavioral, like I said, like if you get startled, you could jump and scream. But physiologically, if you've ever been scared, like really scared, like somebody's jumped out at you and you didn't realize it, or you were actually scared because you were, you heard something in another room and you thought someone was in there and it freaked you out, like your insides react too. Like, or even just think about this on exam day, everyone. Some of you might feel like sweaty, or you might have like a really bad stomach ache because your nerves, right? You're responding to your environment, right? It freaks you out. So we look at this. Yeah, you get that awful stomach feeling. I hate that. It's the worst. But there are a lot of behaviors are there to help favor our survival and reproduction. So we think of parent and offspring interaction. So we have, certain animals that really take care of their offspring. Kin selection is an example. These are all things that you do not need to know as far as memorizing. But if you are given a description of organisms behaving in a way, tie it into natural selection, right? Like that behavior is there for a reason. You might have learned about altruism, which is like sacrificing, like the meerkats, the ones that have to stand up really tall, like they're acting altruistically because they are, they're looking out for predators, but if a predator is there, they're the first one that's going to be taken. So all these behaviors are there to help improve survival of the group. And again, it's just right there. Natural selection favors behaviors that increase survival and reproduction. I'm going to move on from behavior and go into energy. Something that you should know is that all living things require a constant input of energy, but you got to think about what happens if the balance is off. And do all organisms obtain and use energy the same way? Yes, I loved when Taylor Swift put altruism into her lyrics because they were actually accurate. It was pretty good. Okay. Oh, some of you are saying depending on their size, they obtain and use energy differently. Good job. Yes, I love it. Okay, let's get back. So yeah, the answer is no, right? Depending on the organism, depending on the size, depending on where they live, depending on what type of organism. But the one thing they do have in common with energy, if they have a net gain, they're going to either store the energy or they're going to grow. If there's a net loss of energy, that's going to result in a loss of mass or ultimately the death of an organism. So if you ever learned about like the wildebeest in the Serengeti, teachers like to cover that in ecology, and they looked at like the actual bones of dead wildebeest to see if were they starving or were they killed by predators. Energy flow. So different types of organisms use their energy in different ways. So this is something important to know, the difference between ectotherm and endotherm. So endotherms are like us. When you were little, and even people say it now, like cold-blooded, warm-blooded, right? Warm-blooded is what people call endotherms. They use their metabolism to regulate temperature. So if you see in that graph right here, where's my laser? Here it is. This graph right here, the human's body temperature is like staying pretty stable, even if the day goes on and it's hot and cold as the day moves. Whereas a fish, an ectotherm, they have to rely on their behavior because if their environment changes in temperature, they're going to change right along with it. Here's another picture, and I'm going to put a chat. I'm going to put a poll up here. So take a second and read that while I put up the poll. All right, go ahead and respond to the polls. Ready? Mm-hmm. Really nice. So if you looked at the last slide that we covered, the answer is B. They are ectotherms. They likely use behavior, right? And that's a good example of a type of question you might be asked, right? Like you can use data, but then you also kind of have to think back logically. Like, okay, well, the graph doesn't tell me that they're using data, but I can deduce that they have to because I know if their body temperature is changing with the environment, that it's going to have to do something about that it's not going to be able to just handle that up and down. Another thing that we look at is metabolic rate in this indirect relationship where if you have something really big like an elephant, it has a really low metabolic rate, which means it's not burning through its food resources very quickly. Whereas something like a mouse, right, has a really fast metabolism, super fast, just burns right through it. And you just got to think about like, why does that make sense? Like, why does it make sense to have something really big have a really slow metabolism? Like, what would that mean for an elephant for it to have a metabolism that was super fast? And why would that not be a good thing? We're endothermic. Yeah, thank you. Yes. I love what a lot of you are saying. So elephants, I mean, they're big, right? So their caloric intake, their caloric need is probably higher, right, than a mouse. But yes, if they had high metabolism, they'd spend their entire day looking and eating food, right? So it makes sense. Something really, really big shouldn't have a huge metabolism because it would be really energy costly to do that. They'd burn through their energy source way, way, way too fast. And it's more difficult to fill up on those calories. Another thing that we see in ecology, we look at different reproductive strategies, and that's all due to energy availability. So not every organism reproduces in the exact same way. So what I mean by that is there might be some organisms to save energy. They're not just reproducing all the time, right? They have seasonal reproduction. So it's like this one time of year where it's the best to raise their young. So like the wildebeest example, in the Serengeti, they've been able to track that, like, hey, when they're down south where there's not a lot of predators, they're there for like 4 months. And that's where all the wildebeests are having their babies. It's not, there's like this one time a year that they all do it because it's safer, the conditions are best, the grass there is better and more nutritious. And so that's when breeding happens. And then you might also have aseasonal reproduction, which is just continuously. So like specific cues, right? Like rain in the desert, boom, that means it's time to reproduce. And so those are more like a physiological response to the changing environment. We're almost done. You're doing great, by the way. So food chains and energy, I'm not going to go through this very quickly because this is stuff that comes up in like 4th and 5th grade, right? So you've got your heterotrophs and autotrophs. You should know that energy goes from producer to consumer. So your autotrophs are things that can make food themselves. Oops, sorry. And then you should hopefully know the difference between a food chain and a food web. If there's an issue, please let me know and I can add it into tomorrow night. I'm going to skip that. In terms of food webs, I just wrote down some common mistakes that I've seen just from, the past years of grading AP FRQs, that students tend to misunderstand the arrow. Like in a diagram like this, arrows are everywhere. Like this one's going down, this one's going up, and it doesn't really matter. I mean, some of them might be messier than others, but you have to know that the arrow represents the flow of energy. So if I start here, the phytoplankton seems to be the producer because there's no energy going toward it, which means it can do photosynthesis, right? It is being eaten by an herbivorous zooplankton, which is then eaten by this, which is then eaten by this, and so on and so forth. So you can kind of follow through all these different levels. And then just, again, don't assume that the bottom is the producer, and just always double check your data before you place organisms. Your test on Wednesday will likely have several questions about like what would happen if, right? So if less sunlight's available, then kind of just go through that process of, well, what would that mean? Okay, well, that means the plants or photosynthetic organisms couldn't grow as much. If they couldn't grow as much, what happens to the next level? And what happens to the next level, right? And know that changes can disrupt the entire thing. All right, quick look at population growth. You should know the 2 general growth curves here. We have reproduction without constraints, which is exponential. That means there's no limiting factors. This organism can just reproduce like crazy. There is nothing that's going to level it off. This happens a lot with bacteria. Eventually, life happens and constraints are there, rather like if there's room for the organism, is there enough resources? So most things end up in logistic growth where there is some sort of carrying capacity where like, hey, this is as much as the ecosystem can hold for this organism. They're going to level off at some point. And in real life, carrying capacity doesn't look like this perfect line. Carrying capacity is like up and down, right? But it's like the stock market. If any of you ever follow the stock market, you might be following stocks and it's going up and down and up and down. But if you zoom out, it's actually pretty stable, right? So on a small scale, it's like up, down, up, down. But if you zoom out, the up, down, and up, down of carrying capacity is quite stable. And again, carrying capacity is where the population is limited in size. So, that wildebeest population hit 1.3 million and it's not going anywhere. It's not diving. It's not going way up. It's hit a stable population number. And you will do more carrying capacity equations on Monday. Okay, some limiting factors that do keep populations at bay. We've got density dependent, which basically just means the more organisms you have together, the more that factor would affect them, right? So like if a bunch of wildebeests are together, I think that's what these are, and disease comes through, well, if they're really dense, if they're all together, then that disease is going to spread quite large, right? But if it's not a very dense population and they're really spread out, disease isn't going to hurt them as much because they're not close together. It was very similar to when COVID happened. And remember, the cities is where it was really hurting people the most, right? If you lived in the middle of nowhere, your chances of getting COVID were not as bad. But if you lived in downtown Chicago, it was scarier, right? Because it was density dependent and that was what we were living with, right? Some density independent factors would be like, hey, it doesn't matter where what your population is like, if you're spread out or really closed in, it's going to affect you the same, right? So a big drought comes in, bad weather, floods, fire, it's going to affect the per capita growth rate regardless of the density. All right, we are doing so good. We have got 9 minutes. We can do this. All right, so these equations are going to be on the test, right? Mrs. Shoemate will go over them on Monday, okay? And you're going to do wonderfully with them. And if there's still confusion after her night, we will add them to night 5. So Tuesday night, we can always go over math one more time. Okay, I'm going to skip the practice question because we've got to go because we're getting it tomorrow. Last big topic is biodiversity. Now, we think about what diversity is, and it's different, and like having lots of different variations. And so we're talking about biodiversity. We're talking about like all the different species in an ecosystem. So some scenarios that would influence species diversity are going to be changes to the environment, natural disasters, and then large scale changes to a specific population. So for example, if like the keystone species, like the sea otter, if anybody knows about the sea otter, how it's a keystone species where it lives, if the sea otter were to change in terms of like plummet, the entire local ecosystem would fall apart. And so not only are they really cute, but they're a keystone species. So keystone is like, I think it's from like an architecture thing where it's like if you built something up, the keystone is the one that's like holding everything together, kind of like a cornerstone. And if you remove that, the whole thing just, falls apart. And that's what a keystone species is. So make sure you do need to know that. Yes, equations are what will be provided, but something I learned the other day is that you can't like rip out your equation sheet. So you'll have to like flip back and forth. And so just be careful about that. All right. We're doing so great. Okay. A big thing that I want you to know is that the more biodiversity, the more resilient the community is to changes. Just like genetic diversity, right? If you have a population of all green beetles and all of a sudden the green grass turns to, brown, all of them are fair game and they're going to do really poorly. But if you have diversity, if something changes, you have a variety of organisms who can fill a niche. And what I mean by niche, it's Niche. A niche is like an organism's role. Like, are you the top predator? Are you, like one of the main photosynthetic organisms? And so if you want biodiversity where a lot of things can fit certain roles. So we're looking at this. I don't know if your teacher ever uses data nuggets. I love data nuggets. Yes, thank you for spelling it for me. Data Nuggets is out of Michigan State University, and they take real research papers and they make it into a way that is, digestible to high school students. And so these two researchers were looking at Cleveland Metropolitan Parks, and they were looking at the biodiversity there. So they've got Park A and Park B. This is just obviously cartoon drawing, but they considered both species richness, which is the number of different species present. Like, oh, we've got 4 different species total or 5 different types of species. But then they also looked at evenness. So is one species more abundant than others? So you can see here, Park A and Park B, they both have the same species, right? They've got, what is that? Coyote, a deer, an opossum, and a squirrel. They've got the same 4 species, but which one's most biodiverse? And you could do an equation, right? We could do Simpson's diversity equation, and maybe I'll give this picture to Mrs. Shoemate to throw in so you can actually calculate it. But it's gonna be Park B. Yeah, I love it. Great job, everybody. It's Park B, right? They've got a richer biodiversity because not only it's just about like, oh, we've got 4 different species, they have more of each that are equally represented. And so that's going to give them more biodiversity, which means, hey, if something happens in this environment, they're going to be more resilient. Okay, it's time for our practice questions, everybody. We'll just do a couple for Unit 8 because I know my student hosts wants to give you some tips and tricks and good lucks. We're almost done. Unit 8 practice quiz. Let's do this. I'll type it in the chat. Give me a moment. We have 5 minutes, 4 minutes. We can do this. All right, I'm putting it in the chat. So funny. You all love this song. Cracking me up. Okay, let's do it. Ooh, I didn't cover this explicitly. That is though. All right, it's the sun. Great job. Next question. The consumer. Good job. Arrow points to the consumer. Good job. Go. Got this. My goodness. Woo! All of the above. And by the way, I'm laughing at all of you right now because I was like, why are they saying that word in the chat? And it's because of this song, Samosa. That is so funny. Okay, we'll do one more question. I'm laughing. Great job. All right, this question. Population and community are so easily mistaken, but you all, 76% of you got that one right. Population, same species, same area, same time. Community is the next one up, right? So think about when people talk about the community that you live in, like, oh, I live in this community. It's not just people. It's everything in the community. It's the animals that people have. It's the trees in your community. Even when you think about like a community park, the community park has not just one type of tree. It's going to have all sorts of different things. So community is several populations or several different species in a group. So I'm going to end with our quizzes there so that I can honor all of your time and just thank you all so much for coming tonight. I know that our student host wanted to just give you a quick word of encouragement before we leave. So let me just get that over. Hold on. All right, student hosts.
Great. I can go first. First of all, congratulations, everyone. You guys have finished the second day of these cram sessions. You guys are probably tired and even overwhelmed from all the information from today. But we can reassure you that you guys have worked so hard and we are immensely proud of your guys' hard work. It's been a pleasure, honestly, for me. It's been an honor to look at your guys' comments and answer your guys' questions. And we really hope you guys had a good night. And a tip I can say at the end is that keep reviewing up to the exam. However, most importantly, take care of yourself, whether that means doing self-care, getting a reasonable amount of sleep, etc. Your self-care and your personal growth is more, it matters more than a score on an AP test. So definitely take care of yourself, but you guys got this and we believe in you.
Well, you guys made it through 3 hours, so congrats to that. And I really hope that this live session did help increase your confidence for the AP exam. But lastly, as Madison said, take care of yourself. You have to put yourself first. You're more important than any exam.
Amazing. Thank you all so much for being here. I loved it. This was so fun. I will be back here tomorrow night at 7 Eastern to do tons of practice problems. So bring paper, bring pencil, and get ready to do practice. And also just shout out to Jeffrey, who was on the chat and just answering all your questions. They came in just so quickly. And so our host, Jeffrey, was handling that for us. I really appreciate it. And other hosts as well. So thanks, everybody. Have a great night. I'm going to go ahead and stop the stream. Have a great night.